All right, welcome to chapter 3. This is the second of the foundational chapters that I talked about. This chapter is pretty long, and there's a lot of new conceptual stuff in here that might snag you or might hang you up. So, this might be a chapter that you need to be prepared to refer back to in future chapters when some more of this information is brought back up or it's revisited or talked about again. As I've kind of alluded to, this information in this chapter, we're going to talk about it in later chapters, so it's going to be important that you understand this at least to some degree so that we can build upon it later. Okay? So we'll go ahead and get started. All right. So we talked about atoms and all that stuff in chapter 2. In this chapter, we're going to focus on cells. Everything about cells. And you're going to learn a whole lot of different things and new things about cells. This is a very long chapter, so if you haven't already made yourself comfortable, find a comfortable spot. It's going to take a little bit to get through this chapter. So, cells are basically the structural units of all living things, as the slide says. And there's some 50 to 100 trillion cells in the human body. I have no idea how they know that. I feel sorry for the guy that they said, hey, go count all the cells in the human body. I can't imagine what that job would have been like. Obviously that's an about number. So it's, we don't really know for sure, and that's probably changes from day to day. So it'd be tough to get an exact count. Basic structural unit, and of course the basic functional unit of all living organisms is the cell. So that activity of that organism depends on the collective activities of the cells that are within it. So, and this goes into the principle of complementary, is the activities of these cells are dictated by the relative number of their specific subcellular structures. Don't try to get too far off in the weeds on that, but we will talk about the subcellular structures and their importance. And of course, our continuity of life has to do with our cellular functions. So, most cells are composed of 4 different things: carbon, hydrogen, oxygen, nitrogen. Be aware that there are some other trace elements in there too, but these are the main things that make up cells. And they're about 60% water, so there's a lot of water in cells. In general, there's 3 main regions or parts of a cell that we're going to look at, of course, is the nucleus, the cytoplasm, and the plasma membrane. Okay? And that's what this picture kind of gives us an idea of. We'll spend some time talking about all of those things that are, if I can get my markers to come up here. We'll talk about a lot about the things that are contained in here. Little bit about this in here, not a ton about the nucleus. It starts to get pretty complicated when we talk about DNA. We'll spend some time on that cellular, that plasma membrane, so you guys have a better idea of what that's all about too. Okay? The nucleus, of course, is the control center of the cell. Like I said, it contains DNA, and that DNA is needed to build proteins. And it's also necessary for cell reproduction. Absent DNA, there's no way to know what to build the cell out of. So the way I want you to think about DNA, the easiest way to think about DNA is think of it as a very complicated instruction book. If you want to know how to build a model airplane, you get out the instructions and you read the instructions, and it takes you step by step of how to build that. DNA is actually a whole lot more specific in it tells you how to build every part of everything of that cell. So it's not just a matter of finding these pieces and putting them together, it's actually creating those pieces. So DNA tells the cell it holds everything about all the cells, and it's all the information needed to create new cells, also what's needed to create proteins as well. So, 3 regions of the nucleus. We have the membrane or the nuclear envelope, the nucleolus, and chromatin. Like I said, we're not going to get real crazy in depth on the nucleus, but we're going to talk a little bit about it so we have a little bit of an idea. We can see that nuclear envelope that's present there on the outside of that thing. We can see the envelope up in here. It's this outside portion of it. It's what holds it in. And we've got chromatin. We've just got this, it's what kind of the substance that the nucleolus, the center part of that thing, is floating in. It's very important that the nucleus is actually protected from the other parts of the cell, the inner parts of the cell, because it does contain all the building blocks. It's a vital part of the cell. It's the brain center of the cell. It tells everything what's going on, but it does that in code. It does it through the use of codes or chemicals or those types of things. You can also see this blue thing down here that's wrapped around that nucleus. It's called a rough endoplasmic reticulum, and it's rough because if you see all these little dots on the outside of it, these ribosomes that are on the outside of it, it gives it kind of a rough appearance. So that's why it's called a rough endoplasmic reticulum. We'll talk about the reticulum a little bit more when we talk about the specific organelles that are within a cell, and you'll have a better understanding of what its purpose is. But just realize that the nucleus, the nuclear envelope, and all that stuff, and everything that it contains, is the instruction center, or the brain, for lack of a better term, of the cell. Of course, and this is going to go into a little bit more about the nuclear envelope. It's a double membrane, contains nuclear pores that allows for the exchange of material with the rest of the cell. That's important to know, that there is a passageway for communication between the nucleus and the rest of the inside of the cell, and that's through the use of those pores. And then, of course, there's this jelly-like fluid that's inside of there. It's called nucleoplasm, that's inside of that. There's nucleoli, which are sites of ribosome assemblies, meaning it's places where ribosomes are put together. And then they migrate into the cytoplasm through the pores to serve as a site for protein synthesis. Just realize that ribosomes are where proteins are used to create proteins, to build proteins, and proteins are used for other purposes, a wide variety of purposes throughout the body. But ribosomes are what does that. I don't think it'll let me go back. Yeah. Remember I told you these little dots here that we were looking at on these little dots here? These are ribosomes. There's also ribosomes present on the nuclear envelope, and that's where protein synthesis occurs. So that's vital to the survival of any cell, is the ability to create proteins in order for the cell to keep going and doing its thing. So chromatin is composed of the DNA of DNA and protein. It's present inside that nuclear envelope when the cell isn't dividing, and it's kind of scattered throughout the nucleus. It can condense, and you've probably heard it referred to as chromosomes, and that's when that cell becomes the chromatin condenses to become a lot more dense, and it creates those rod-like bodies. Those rod-like bodies are called chromosomes, and that's right before the cell divides. Like I said, I'm not going to get real crazy into the function of the nucleus, because for the most part of what you guys will be studying at a later date, it doesn't talk much, it doesn't help to know a whole lot more about the nucleus, and I'm afraid it would just kind of cloud things up for you if we went crazy in depth into that. I'd rather make sure that we're focusing our time and attention on those things that are a little more vitally important. But I do want you to have a familiarity with that part of it. Okay? All right. Plasma membrane. This is something that we need to know quite a bit about, and need to understand it. It is the transparent, it's that barrier outside of the cell that surrounds the contents and actually separates the cell from the outside from the rest of the world. It's kind of like the walls in your house. If we use that as an example, it's what separates your house from the outside world. It's vitally important that cells maintain their own internal environment, otherwise they wouldn't survive. Much like your house, it's kind of tough to, manage the temperature inside of your house if it doesn't have walls. Same way with cells. That plasma membrane is very important to doing that. It does contain some cell contents, and it separates, like I said, from the surrounding environment. That plasma membrane is made up of several different things. One of them, and the chief among them, the main one among them, is those phospholipids, which we'll look at a picture of it here in a minute. There's some cholesterol, proteins, even some sugars present there on that plasma membrane, and all of them serve a vital purpose. So if we look at this picture of this plasma membrane here, the first thing that we look at is this phospholipid layer, and that's this right here. If we just looked at these little guys right here that are shaped like, I don't know, just like a pair of legs and whatever it is, I don't know. But if we look at those, remember we talked about those in an earlier chapter and how those make up a cell membrane. Remember that they have hydrophiliac heads, meaning heads that like water, the heads of them like water, and hydrophobic tails that don't like water. So if water gets in here in the middle, it's going to be pushed out to either the inside or the outside. Now, these are pretty tightly bound together so that it actually creates a barrier, and it kind of keeps water and stuff out of it. However, water can still pass through, and some things, some solutes can still pass through that barrier. Other things are going to be too big to pass through in between these, and we're going to have another way to get them through there. But this phospholipid bilayer that we're looking at here actually separates the cell from its internal from external environment. Okay, and then we've got some proteins in here. Here's a little glycoprotein that's pointing at there. These are some other proteins in here, and they have different purposes. And you can see this channel through the middle of this protein. So that channel in there has some sort of a purpose. Like I said, it's probably going to allow something to pass through that membrane that couldn't pass through the bilipid layer of that membrane. Also, we've got these little sugar groups up here. You may be wondering why that sugar group is up there. Some of those sugar groups are actually designed to identify that cell as that it's belonging, that it's supposed to be there. Because if we have another cell from the immune system come by and make contact with this right here, and it says, hey, look, I don't recognize you, you don't belong, that immune cell will actually destroy this cell. So it needs to be able to communicate who it is and where it belongs to the outside, to those on the outside. Also, if this cell is, say, a liver cell and it winds up floating somewhere around the pancreas, the cells, the immune cells around the pancreas are going to go, hey, look, you're a liver cell, you don't belong here, and it'll get destroyed because it's in the wrong place. That's what a lot of these sugar groups or these glucose groups are designed to do. They're a part of the communication system of the cells within the body. So remember those molecules that I talked about, the polarity of those phospholipids, those the hydrophobic heads or the hydrophilic heads and hydrophobic tails. Just remember that. You need to hang on to that little tidbit of information. Like I said, hydrophiliac, water-loving, hydrophobic, doesn't like it. Whoops, sorry. So the tails are going to push water away. The heads are going to pull water towards it. That's going to keep the water from building up in the center of that membrane, that double-layered membrane, and it's going to keep the water. It's going to allow for water to stay on the inside and the outside of the cell, just kind of how we're made. And that's just repeating that same thing that I said. And just excuse me if I kind of move through a little bit fast, because there's a lot of material that I'd like to cover, and I want to try to move through this fast so you don't fall asleep on me while you're sitting there. You may have already fallen asleep. If you have, wake up. I'm kidding. I will try to move fast. That way it's maybe not too painful. So there's also proteins present in that membrane, and they're responsible for some specialized functions. They may produce some enzymes. They may be a receptor to sense other things that are outside going on in the environment. And as I kind of alluded to, some of them are transport channels or carriers that will carry objects or substances or solutes that are too big to pass through the membrane. They'll carry them through that membrane manually. Basically, just grab onto them and pull them through. Remember, though, that does take some energy to do that, and we'll talk about how that energy works or where it gets that energy from here in a little while. The sugars that are present, they some of them, like I said, are used for identification purposes. Some of them actually can make a cell be able to hang on to something. So they have some different purposes. Membranes can be bound together and held together in 3 different ways. Those 3 ways are through those glycoproteins that act as an adhesive or kind of glue them together. Some of them have kind of a little tongue and groove way that they hang on, and some of them have these specialized junctions called gap junctions to where that they can communicate and hang on to each other. Depending on the type of bond that they have will kind of determine how well those cells kind of stick together and how well they can be pulled apart. Some of these, one of the junctions is called a tight junction. It's basically impermeable. This is the kind of junction that we typically find, for example, on our skin. It's an impermeable junction that doesn't allow things to pass through it easily. It creates leak-proof sheets and doesn't allow things to pass through the space in between those cells, so it's kind of locked out. It's got it locked down. Desimones, I don't know if I'm saying that, desimosomes are the anchoring junctions that prevent cells from being pulled as a result of mechanical stresses. So it's kind of a fibrous way that cells hold on to each other, so that instead of just the cell membrane sticking together, these desmosomes, they actually penetrate into the inside of the cell, kind of like finger or hair-like projections, and hang on. And that's where we see a lot of stresses in the body, to where they provide some anchoring points to hold those cells together. And of course, gap junctions are primarily used as communication routes between cells. There's a hollow center in those in the center of those proteins that span the width of the 2 adjacent membranes, and they allow communication within the cells in order for it to perform whatever action it is. We see this primarily or mainly in cardiac cells. Cardiac cells have the ability to communicate rapidly with one another because of these gap junctions that allow that when we stimulate one cell to contract, it quickly spreads to all the cells of the heart, which is convenient because we need all the cells of the heart to be able to squeeze at the same time in order to perform the function that they do. That's where the gap junctions come in and make that possible to do that. So we can see an example of some of these connections. We see gap junctions down there at the bottom. We see these gap junctions here that I was talking about, and you can see to where that they literally connect one cell with the other. The desmosomes right here, you can see the finger-like projections that go to the inside of that cell, and the way that they bond here in the middle creates a really, really tight junction, or a really, really tight anchor, excuse me. And then the junctions, the gap or the tight junctions up here at the top, these are mainly because of the stickiness of these sugars that are on the surface. They help with that stickiness where that they kind of adhere together. What they do though is you can see how abundant that they are. These would run the full length of the cells that they're adjacent to and create a barrier to where that nothing could get through there. You can see how these are kind of have that stitching look that kind of creates a barrier. These down here, on the other hand, don't create a tight barrier. These do kind of create a tight barrier, but they have a different purpose. So that's where we see some of those differences. Okay, there we go. The cytoplasm, the substance inside of the cell, but outside of the nucleus of the cell, that jelly-like stuff inside of a cell, is called the cytoplasm. And it's where most of the cellular activities take place. It's also where what we call the organelles are located. Organelles is basically just small organs. If you try to think of it that way, you've got organs in the bodies. And the small version of an organ is an organelle, and that's what we find inside of the cells. Okay? So that cytoplasm has three major elements. It's got cytosol, which is that fluid that it suspends the other elements. It's kind of a thicker jelly-like fluid. Also, like I said, the organelles, it's that machinery, the internal organs inside of that cell that actually do the work. And also it's got something called inclusions, or chemical substances that are stored nutrients inside of those, and the nutrients and things that are inside of that cell. So it's got like a storage capacity of some substances that cell might need to hang on to and utilize for future reference. So when we look at this picture here, we see the all of these, and this little cell they've got here, I got to tell you, it is just cram-packed with organelles and things that are going on in here. But if we want to try to find if I can get my deal to work here, if we want to try to find the plasma inside of here, then we're going to find it. We can see it's just this jelly-like substance that's around here. But there's a lot of things that actually are holding this cell together. We've got these centrioles here that are kind of helping hold the cell together and kind of helping create a shape to that cell. And we've got some cellular activities and things going on over here. So just a picture of a cell. Realize the picture of the cell that we're going to use is just kind of a standard picture of a cell. Kind of like we talked about with the atoms, it's kind of like a standardized picture that we use. Cells do come in many different shapes and sizes, and when we get to the end of this chapter, we'll briefly talk about some of those different shapes and sizes that they come in. So, organelles, this is one of the places that we'll spend a lot of time talking about the different things that these different little organs inside of the cells do. But they're specialized cellular compartments is what they refer to them as. A lot of them are actually membrane bound, meaning they're within a membrane inside of that cell because of the things that they're doing requires that they be able to do them absent the internal environment of that cell, to protect the cell or to protect the process of what's going on inside of that organ. That's the way that it's able to do many of those specialized functions. The first of the organelles that we're going to talk about, and you've probably heard of this one before, it's always referred to the powerhouse of the cell as the mitochondria. But what I want you to realize is some cells have hundreds or thousands of mitochondria in them in order to create energy. Okay? So it says it's the powerhouse of the cell, but what it's really doing is it's creating energy, and the energy that it's creating is in the form of ATP. So adenosine triphosphate is the energy that it's creating. These little guys will change shape continuously because of the work that they're doing. It has a double membraned wall with it's called a cristae on the inside of the membrane, and they carry out reactions where oxygen is used to break down food. Now, it creates ATP, or ATP is created within the mitochondria, and that ATP is what is used for cellular energy. The creation of ATP requires oxygen and glucose in order for that to be done in an efficient and effective way, and that's where that takes place. So energy production, if you want to know all this oxygen that we breathe in and why oxygen is important to our body, and if we don't have the right percentage of oxygen coming to our body, why that's important, it's because the mitochondria need oxygen to produce ATP. Without ATP, the cells can't perform the functions that they're designed to do, and then we run into big problems. So just remember, mitochondria, powerhouse, ATP, needs oxygen and glucose. Okay? Ribosomes, on the other hand, are those bilobed dark bodies, as it calls them, but it's those little dots that we've seen on the outside of the nucleus, and we've seen it on the endoplasmic reticulum. That is why we call it a rough endoplasmic reticulum. This is where protein synthesis is done. And also it says ribosomal RMA is made there too, but mainly what I want you to think of that as is protein synthesis. It's where proteins are made. They are in two locations. Some of them are floating free in the cytoplasm, and there's others, the rest of it's on the rough endoplasmic reticulum. It's just where they're located, and that's where proteins are made. Then we have endoplasmic reticulum. There's two different types. We already talked about the rough. Remember, the ribosomes that are present on the endoplasmic reticulum is what makes it rough. But there is a type that's called smooth endoplasmic reticulum. Just realize on smooth endoplasmic reticulum, no proteins are made because protein synthesis happens with ribosomes. Absent ribosomes, no protein synthesis. So endoplasmic reticulum, rough and smooth. Rough has ribosomes and creates protein. Smooth does not have ribosomes and does not create protein. So, and that's what this slide is talking about here. Also, on the rough endoplasmic reticulum, where those ribosomes are present and it's creating proteins, there's usually, well, what typically happens in order to move the protein within the cell. So let's say that a cell creates a protein, but that protein is not going to be used by that cell. It's going to be used by some other cell within the body. Then what happens is that cell will create that protein. It'll put it in a vesicle, or it'll basically put a membrane around it and transport it through the middle of that cell to the outside of the cell where it's kicked out into the tissues or the bloodstream or wherever, and that protein can go do the job that it was intended to do. Now, some proteins are actually used within a cell, and they may be still put in a vesicle or in a membrane, in a little membrane pouch, and carried to another organelle within a cell. But typically, those proteins are going to be traveling inside of some sort of a vesicle. And when I say a vesicle, it's like putting it in a little Ziploc bag to carry it across, the room so that you don't lose any of it or you don't contaminate it, basically. And that's why it's carried in a vesicle. Some proteins can actually be harmful to that cell in particular, so we don't want to turn them loose there. We need to send them where they need to be to where that it's safe. And of course, the more a particular cell is responsible for creating and exporting proteins, is the more abundant endoplasmic reticulum that it'll have. So just realize that that's where proteins are made. And then we can see, this kind of gives an example of it. We got one of these proteins being made inside of that cell. Proteins being made here inside of that cell, and this down here is the endoplasmic reticulum, and this is a ribosome on top of that. We'll talk about this RNA that's going on up here, this little RNA that's going up here, but it's creating a protein. That protein is, once it's created, it is then packaged by the endoplasmic reticulum. It pinches off part of the membrane on the outside, and it creates this vesicle to transport it in. Think of the reason why that it needs to transport that in a vesicle is, let's say you need a cup of bleach to do laundry, but you keep your bleach at the other end of the house, from where the laundry room is, and you need to get the bleach from the other end of the house to the washing machine. But how are you going to do that? You can't take the bleach container down there because it's just not possible. So, and if you poured it in your hands and tried to carry it through the house, you're gonna stain the carpet, you're gonna stain yourself, and that kind of stuff, and it's not safe. So what you could do is you could pour that bleach in a Ziploc bag and safely carry it from one end of the house to the other, pour it into the washing machine, and everything would be fine. So that's what the endoplasmic reticulum is doing with proteins, because some of those proteins may, in fact, not be safe and could be harmful to that cell. Okay? And this is just going through that. There you go. Smooth endoplasmic reticulum functions in lipid metabolism, and it also works to detoxify drugs and pesticides from within the cells. So rough endoplasmic reticulum is primarily designed for protein synthesis. Smooth metabolizes lipids, which is an important thing, and detoxifies the cells, which in turn detoxifies the whole body. The Golgi apparatus, this weird looking little stack of flattened membranes, it basically modifies and packages the proteins coming from the rough endoplasmic reticulum via those transport vesicles. So let's say we've got that same cup of bleach. Now let's say, hey, your mother-in-law needs that cup of bleach. So you go back to that back room, you put it in a Ziploc bag, zip up the bag, you carry it through the house, and you get to the back door and you think, wait a minute, this bag is not strong enough to carry this bleach across town in my car, so I need to put it in something else. So you grab a mason jar and you put the Ziploc bag inside the mason jar and close the lid on that. Now you've got a good solid transport vessel to where that you can transport it without spilling it in your car and bleaching your seats or anything like that. So that's what the Golgi apparatus does. It basically takes those proteins and packages them together. Now, while those proteins are in there, because it is a membrane, so it can take the proteins in, and it can combine proteins with other proteins or other things together, and proteins and lipids and that kind of stuff, and then ship those out, repackaging them and ship them out. So, and here this slide alludes to the fact that it produces 3 different types of packages. Vesicles that it kicks out. Like I said, it's kind of like us putting the bleach in the mason jar and taking it outside of the house, or things that are done inside of there with proteins and lipids. It may package them and so that it can be carried even safer than in the Ziploc bag across there. And of course, lysomes that are actually protectors inside of the cell that get rid of things that don't belong there. So, okay, so usually we'll see a Golgi apparatus that is sitting pretty doggone close to the rough endoplasmic reticulum, and that's this little squishy looking green guy right here. And you can see it looks like a stacked membranes or stacked plates. You can see the rough endoplasmic reticulum here creating these vesicles of proteins that are then taken up by the Golgi apparatus. They come on through this thing. They're repackaged, reshaped, reconstituted, put in another vesicle, and kicked out. This one may stay inside. This is a lysome, actually. It's going to stay inside and be utilized to deal with other substances that may not belong inside of the cell. This path right here is going to go to the outside of the cell. Actually, this path is going outside of the cell too. This one may be reclaiming some. This one over here may be doing some reclaiming. But we can see how this one is going out and around, and then this opens up here and allows what its contents to come out. Some of them may actually come out of the cell in a vesicle too. It's possible for that to happen as well. But that's what the Golgi apparatus does. It's the packing and shipping department. It's kind of like the UPS of the cell. It's where everything's packaged and shipped. Okay? So lysomes are those, remember those lysomes that were packaged by the Golgi apparatus, those stay inside of the cell, and they contain different enzymes that are produced by those ribosomes. The thing is, what these enzymes can do is they can digest worn out and non-usable cell structures within the cell so that they can be recycled or gotten rid of. It's the housekeepers. It keeps things clean, and it keeps things from building up. It also helps with bacteria and other cell debris and that kind of stuff. These lysomes are vitally important to the survival of those cells. Okay. So peroxisomes are membranous sacs of oxidized enzymes, and they also do some detoxification and clearing out of stuff. But mainly what they do, they deal with alcohol and formaldehyde. They break down those free radicals, or they break down free radicals and typically convert them to hydrogen peroxide and then water, and then get rid of them out of the body. So they're another one of those protective measures inside of the cell that keep the cell from being damaged by the things that are inside of it, or even some of the waste products that are created by the cellular production of the other things that the cells create. Basically, and what's interesting is they replicate just by pinching in half, or attaching to the endoplasmic reticulum, and just create more. Also, there's a cytoskeleton present inside of there, and it's typically considered an organelle. Basically, it's a network of protein structures that are all within the cell to help keep that cell help that cell keep its shape, or keep it within its framework of a shape. And there's 3 different types of elements that make up that cytoskeleton. Of course, there's microfilaments, intermediate filaments, and microtubules that make up all of those elements that are part of that cytoskeleton. Okay? All right. So, and we can see those. This is a pretty good depiction of those. And this one up here where it talks about the microfilament, where it talks about actin, we're actually going to talk about actin a little bit more when it comes to microfilaments of skeletal muscles. We'll talk about that microfilament, and I believe even myofilaments. It's actually what it'll be called as muscle because it's myo. Centrioles are these rod-shaped bodies that are made of microtubules. Remember, the smallest unit of that cytoskeleton. And these centrioles actually generate those microtubules, and they direct the formation of the spindle during division. So they're a key player in cellular structure. All right, remember these there's some surface extensions that are found on some of the cells. Now, not necessarily like the ones that I told you that were the identifiers of a cell, but some of these have projections that stick up. One of the first one that it has up here is cilia, which move materials across the cell's surface, located in the respiratory systems to move mucus up. Your lungs naturally and normally produce a small amount of mucus intentionally. It's designed to trap foreign substances and pollen and all that kind of stuff that we breathe in. And those cilia will do this wave-like motion to kind of push that stuff up towards the top. They'll push it up so that we can cough it out. Of course, smokers, they begin to, with smoking, actually damage that cilia to where that it doesn't work as well. And they wind up having more mucus build up, not only because of the smoking, but because of the poor ability for the cilia to be able to work. The other type of surface extension is the flagella, used to propel a cell. There's only one cell, one human cell that is capable of doing that, and it's the sperm cell. It's the only one that is capable of propelling itself. And of course, there are another thing called microvilli, which are these little finger-like projections of the plasma membrane, and they're designed to increase the surface area for absorption. We usually find those located predominantly in the digestive tract. So it's these little finger-like projections, millions of them on the top of a cell. And that's the only one that picks, of all cells that it picks that one to give us a picture of. So the human body has over 200 different cell types. And cells are going to vary in length from 1/12,000th of an inch to over a yard. So, like I said, don't think of all nerve cells as being of all cells looking the same. Cells are different. They're different shapes. They're different sizes. They're different lengths. And they all have a specialized function. And that's where they get their shape. Their shape is determined by the function that they do. So that's why we have cells of different shapes. And we have cells that connect. Their primary purpose is to connect, such as fibroblasts, which secrete cable-like fibers. Erythrocytes, which are red blood cells that carry oxygen. Interesting thing about erythrocytes is they don't have a nucleus, they don't have mitochondria, they don't have a lot of those things. They don't have much of anything that any of the other cells have, but they do have what's called hemoglobin that allow them to carry oxygen. When we get to that chapter, we'll talk a whole lot about how that works and what all that involves. But we can see these fibroblasts here on the left, and you can see their purpose. They're actually holding things together. They do have a nucleus and some endoplasmic reticulum, Golgi apparatus, and that kind of stuff. But then we look at the red blood cells on the right, and they have nothing. What's interesting is they do have a very strong, rigid cellular structure internally that helps them hold this disc-like shape. This disc-like shape is actually in order to increase its surface, increase its surface size, but also allow it to be able to pass through some very, very small blood vessels to get squished down, and it also enables it to be able to take a beating. Red blood cells take a beating in the job that they do. All right, so we also have epithelial cells, and these are in the category of cells that cover or line the body organs. These are sheets packed together, sheets of cells packed together, and they have those intermediate fibers that resist tearing during rubbing and pulling. One of the things that I always like to point out here, and I'll probably bring it up again when we talk about the digestive system, is remember that even though we take food, eat or drink something into our body, it is still outside of our body because of this this lining, these membrane linings that actually separate our body from the outside world. So even though we consume something, it is still exterior to our body tissues. Okay, so it's a little different. It's a little tough conceptually to sometimes to kind of get past, but start trying to kind of think that way a little bit and realize, yes, I know I just ate a taco and it's inside my body, but it's not really inside my body. It's not in my tissues. If we drew your blood, we wouldn't find, taco sauce. So it's not really in your body. It's in your digestive tract, which is still outside of your body, technically speaking. Okay? So we can see these epithelial cells here and kind of how they're held together by those junctions. And then we also have some cells that move the body, move organs and body parts. Skeletal and smooth muscle cells are both the ones that we talk about, and also cardiac cells too. These have specialized filaments in them that allow them to shorten up forcefully and change their shape dramatically so that they're able to move body parts or do particular things. Well, on the left we can see a smooth muscle cell, and the one thing that I want you to notice is to see the lines on, a cardiac muscle cell on the left. If you can see these lines that are present on that, these little lines going this way, they're called striations. So we call skeletal muscles striated muscle cells, and we can see over here there's no striations on these cells, on the smooth muscle cells, so we call them smooth. Actually, cardiac muscle cells do have some striations, but because they're so unique in what they do, we call them cardiac muscle cells because they are very different from skeletal muscle cells. Okay? We'll talk about each one of those much more in depth at a later date. So don't try to think that, oh my gosh, I don't have enough information to understand these. Believe me, we'll get into them pretty deep. There are cells that actually store nutrients, believe it or not, and fat cells are the ones that do that. Basically, they're lipid droplets that are stored in cytoplasm. So some of us are much better at storing fat cells or storing lipids than others. I happen to be an expert at it. So I am great. I've got that down to a fine art of being able to store lipids in the form of fat cells. Interesting thing, if you've ever asked yourself why are there more obese people in our country now than there were, say, 30 years ago? I don't know, many of you might not have been here 30 years ago, but 30 years ago, I distinctly remember there were very, very few obese people in this world, and even fewer morbidly obese, 600-700 pound people. That's become so normal now to see people that are overweight that we don't even think anything of it. Well, it has to do with our diet. The food producers have gotten so good at producing food that is so nutrient rich for our bodies, and that's in part what our mouths crave. that we wind up with a whole lot more lipid, a whole lot more fat, a whole lot more garbage that we shouldn't eat that winds up getting stored in fat cells. At least that's who I'm going to blame it on. I'm not going to take any credit for it. It can't be my fault. So there's also some cells that fight disease. One of them is a macrophage. Basically, it digests infected microorganisms. It just goes in and devours them and destroys them and just deals with them in that way. And there's a picture of the little guy. So there's also some cells that gather information. These are very specialized cells. Nerve cell is an example of that. It can both transmit and receive, but the same nerve cell can't transmit and receive. It has to be two different nerves transmitting and receiving. So the nerve that lets you know that you stepped on a thumbtack is different from the nerve that makes you pick your foot up. So completely different nerves. One's sending, one's receiving. One nerve can't do both. It can, but not in that way. Which we'll talk about a whole lot more when we get to the nervous system. So, and of course, reproductive cells, oocytes for females and sperm for males. Oocyte is the largest cell in the body. Interesting, isn't it? Wouldn't have thought that, but it is. And of course, it divides to become an embryo. Sperm is a specialized cell, so it can get to that oocyte and create an embryo, play its role in creating it. And yet again, this picture of the sperm cell. I don't know what the fascination is, but these were not my slides. I did not put them together. I'm just using them. I did add some stuff to them, but I didn't think to delete stuff. Cells have the ability to metabolize, digest food, dispose of waste, reproduce, grow, move, and respond to different stimuli. And we're talking about in a general way. We're going to have cells that are from different parts of the body that may not be able to do things as well, or may not be able to do some of these things, like red blood cells. Red blood cells can't do a lot of this, or maybe not any of it. So just realize we're talking about that in a general form. Membranes need to transport things, and they need to be able to move things in and out. And membranes are completely reliant and dependent upon balance and everything being equal. And that's what they try to do. When we have a mixture of two or more components, we call that a solution. We can dissolve that solution by adding a solvent. We can add a medium. Typically we use water, and that's what's most present in the body, and that's the universal solvent that our bodies use. And of course, components that are in small quantities within a solution are called a solute. So you go to the restaurant, you sit down, you order you a glass of tea, and you grab the sugar and you start pouring sugar in there. Now your tea has the solute. Not only does it have tea in it, but it has sugar. So that sugar becomes a solute once you've put it in that solution. Okay? And if you just had a cup of sugar and you wanted to dilute that sugar, you would add water to it in order to dilute it. So you kind of see where I'm going. So, and if we mix two or more components, our sugar and our tea, we actually come up with a solution. So when you mix your tea at the restaurant, you've created a solution. So, all right. So there's fluid inside of the cell. We've talked about that neoplasm and cytosol that's inside of the cell. And that solution that's inside of the cell, it's called a solution because it has gases, nutrients, salts, and all that stuff that's dissolved in water. So there is actually a solution inside of the cell. Outside of the cell, there's fluid and all that stuff out there too. All kinds of different ingredients, such as nutrients, hormones, neurotransmitters, salts, waste products, and all that stuff. All that outside of the cell. But the two are isolated. That cellular membrane keeps the two isolated. Now, there is a limit to what that cell membrane can do. It's not perfect. So there can be some problems. While it is selectively permeable, meaning it'll only allow certain things to pass through. Remember I said only things that are small enough to pass through that membrane can just filter in easily. The problem is, or the good thing is other things have to be transported in. We need proteins to actually carry them in and out of the cell membrane. But that permeability of that membrane, we call that selectively permeable, meaning some things can come through easily, other things are going to require effort and work in order for that to come through. And many of the undesirable substances are managed to be able to be kept out. So mainly that membrane is permeable to the things that are really safe for it and relatively harmless, but it's not permeable to those things that can be damaging to it, if that makes sense. However, like I said, it's not a perfect world, right? So there are two basic methods for moving a substance in and out of a cell or in and out of a cell through the membrane. There's two basic ways to do that. The passive process that we've talked about just a little bit, it's the one that doesn't require energy. So if something can just kind of slip through the cell membrane, it doesn't require energy, it doesn't require ATP, so it's passive. The active process is basically anything that there must be energy expended in order to get that substance either in or out of a cell. So if there's a protein that is required to grab a hold of a substance and pull it in, or grab a hold of a substance and push it out, that's going to require energy in the form of ATP, and that is an active process to move it across that. I'm going to see if I can get this to play. Well,
in order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion.
We'll talk a little more about passive transport or diffusion here in a minute.
Bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump. But because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins,
Okay,
from the
I'm going to try to make this thing stop it there. All right, so I know it talked a lot about different concentration gradients and all that kind of stuff. So I don't want you to try to worry about that too much. We're going to talk about that. We're going to talk about those concentration gradients and diffusion and all that stuff much more in depth here in just a minute. I just wanted you to realize that those two paths that it was showing were that those substances are moving across a membrane. You know, that was the main thing that I wanted you to understand. So, all right, let's move on. All right, diffusion, basically, and I know that video just talked a whole lot about diffusion, and it would have been nice to know what diffusion was beforehand, but basically, just realize that particles have a tendency to distribute themselves evenly within a solution, and that's what we call diffusion. So the best way that I describe it, if I go back to that glass of tea reference, if you pour that sugar in your tea and you stir it up, that sugar becomes distributed evenly throughout the entire glass of tea. If you pour more in there than what can be evenly distributed, it'll collect down there at the bottom. Look at your kid's glass of tea, or remember the glass of tea that you had when you was a kid, and you'll know what I mean. You know, there's an inch of sugar in the bottom, and it's settled down in the bottom because it can't be suspended within the solution. It can't diffuse any more sugar into the solution. It's done all it can. Basically, what causes this to happen, how this works, is through kinetic energy, and it's that energy in motion that causes those molecules to move about randomly. It's a whole science-y chemistry kind of thing to understand that kinetic energy that's there all the time. But just realize diffusion is that dilution down, that evening out of all the molecules to where that they balance out. And what will happen is we'll have a molecule move from an area of high concentration to an area of low concentration, or what's referred to down the concentration gradient. So if we had a really, really sugary glass of tea and a glass of tea that didn't have any sugar in it, and we were able to just kind of put them next to each other and push them together very quickly, both, all of that would become evenly the same sugary amount, basically. Or here's a better example. You get your and I don't put sugar in my tea, so I know I keep using that reference, but I don't drink sugared tea. But let's say you get your tea. I do drink sugar in my coffee, so maybe that's a better way. So let's say you get your coffee just perfect, the right amount of sugar in your coffee, and you get it drink it down to just below half, and you're talking to whoever you're eating dinner with, and the waitress slips over there while you're not paying attention and pours more coffee in your coffee cup. Now what happens when you take a drink of your coffee? It doesn't taste as sweet, right? Is the coffee sweeter at the bottom, or is it just, and is it just less sugar at the top, or what? Well, no, that sugar moved from the area that had, the coffee that was already in your cup that had a greater concentration of sugar in it. It quickly distributed to the coffee that was just poured in your cup, so that all of it is even now. So now you're going to have to add more sugar because we diluted it. That's the only way that you're going to fix that. So diffusion is just a diluting down. So it's trying to balance. It will always try to balance out, and that's what diffusion is trying to do. And sometimes that's dependent upon temperatures and molecule size and that kind of thing. Of course, smaller molecules are going to move faster. They're going to diffuse faster. Warmer molecules will move faster as well. So there's some things that kind of affect that. A perfect example is if you're cooking in the kitchen and you're cooking something and you're going to add sugar to it. Some recipes call for adding sugar to while it's hot because the sugar will distribute faster. So we can warm up that molecule and make it move faster. Sugar is actually a very large molecule. It doesn't move very fast. That's why whenever they bring you your coffee or your sugar, or I'm sorry, your coffee or your tea, and you put sugar in either one of them, they give you a spoon because you're going to have to stir it up. You're going to have to mix that substance up with that sugar in order to get the sugar to distribute faster. Otherwise, you're going to sit there for a very long time waiting for the sugar to distribute by itself by its own diffusion. So remember, just so you understand, diffusion is that movement of the concentration of a substance. And looky there, it gives us a cup of coffee and drop it in a sugar cube. You're going to have to leave it setting all day. It will taste sweet by the end of the day because the sugar molecules are going to move really slow. They're a big fat molecule. And incidentally, sugar molecules do not pass the membrane of a cell quickly, or they can't at all. They have to be facilitated to get in there. It takes energy to get a sugar molecule into a cell. Can't just come across super easy. So here's an example of diffusion. We drop the sugar cube in the beaker on the left. We wait long enough, and what we get is what's in the beaker on the right. That's going to be completely dependent upon what substance it is that we dropped in there. Some move a little bit faster. Salts would move a little bit faster, and actually a lot of salt would already be distributed before it ever got to the bottom. Salt is much smaller. Sodium chloride is much smaller than sugar, so it would move a lot faster. So they'll move by diffusion when if any of the following things apply. If they're small enough to pass through the membrane. So when we're talking about diffusion across cell membranes, so if we put a cell in a glass of water, and we change what was inside of that water, those molecules would actually either move, or the water would pass through that membrane. So it all just depends on whether that molecule is small enough to pass through the membrane, the pores of the membrane, or if the molecules are lipid soluble. If they're lipid soluble, they will, I believe they'll pass. I'm getting mixed up here. Or if the molecules are assisted by a membrane carrier. Lipid solubles are actually tougher to get through the membrane. That's right. And of course, if we have to have a carrier carry it through the membrane, it's going to take longer because it's going to, basically, if you've ever been standing in line, to exit the movie theater, you've got to wait because there's only a certain number of doors that you can go out. So it's kind of that same principle. You've got to wait until there's an opening for you to go out, and that's what the carriers have to do. So it affects that diffusion of substances across that. We call it simple diffusion if the process is unassisted. Solutes, okay, are lipid soluble. That means they're small enough to pass through the membrane. So lipid soluble, I was afraid I was getting that wrong. Lipid soluble substances can pass through the membrane easily, and don't require energy. So it's just simple. They just diffuse across there. Very simple to get across. And you can see an example of that. Lipid soluble solutes that are outside of the cell diffuse across into the cytoplasm really easily. Easy process. Osmosis is another type of diffusion, but osmosis has to do with the water side of things, and not so much the solutes. Solutes play a role in it, but it has to do with the movement of water. So osmosis is the diffusion of water. Some high polarity water molecules easily cross the plasma membrane through aquapores. Water can move up and down, or can move down that concentrate. But anyway, basically what I want you to realize is osmosis is the process of water moving across. Now, there are some solutes that we may have that can't cross that membrane easily. So the only way to balance out, the cells always want to try to balance out what's inside the cell with what's outside the cell. Sometimes the only way to do that is to move the water, because it would take too long to move the solute, or it would be an active process to move the solute to balance it out, or the solute that's there, it may be too dangerous to have that concentration of it inside of the cell. So it uses osmosis to do that. Here's a video from Khan Academy. I plugged these in. They're the same ones that it shows there in your in the list underneath this underneath chapter 3. So it's not any different. It's the same video. I just plugged it in here so it fits in the process really well. We'll just watch it. I'll stop it if it gets to where we need to be.
In this video, I want to cover several topics that are all related. And on some level, they're really simple, but on a whole other level, they tend to confuse people a lot. So hopefully we can make some headway. So a good place to start, let's just imagine that I have some type of container here. Let's say that's my container. And inside of that container, I have a bunch of water molecules. It's just got a bunch of water molecules. They're all rubbing against each other. It's in its liquid form. This is liquid water. And then inside of the water molecules, I have some sugar molecules. Maybe I'll do sugar in this pink color. So I have a bunch of sugar molecules right here. I have many, many more water molecules, though. I want to make that clear. I have many, many more water molecules in this container we're dealing with. Now, in this type of situation, we call the thing that there's more of, the solvent. So in this case, there's more water molecules, and you can literally just view more as the number of molecules. And I'm not going to go into a whole discussion of moles and all of that, because you may or may not have been exposed to that yet. But just imagine whatever there's more of, that's what we're going to call the solvent. So in this case, water is the solvent. And whatever there is less of, so the more, water is the solvent, and in this case, that is the sugar, that is considered the solute. This is the solute. So the sugar, it doesn't have to be sugar. It could be any molecule that there's less of than the water in this case. Sugar is the solute. And we say that the sugar has been dissolved into the water. Sugar has been dissolved into the water. And this whole thing right here, the combination of the water and the sugar molecules, we call a solution. We call this whole thing a solution. And a solution has a solvent and a solute. The solvent is water. That's the thing doing the dissolving. And the thing that is dissolved is the sugar. That's the solute. Now, all of this may or may not be review for you, but I'm doing it for a reason, because I want to talk about the idea of diffusion. Diffusion. And the idea is actually pretty straightforward. If I have, we'll say the same container. Let me do a slightly different container here, just to talk about diffusion. We'll go back to water and sugar, especially back to water. But let's say we have a container here. And let's say it just has some air particles in it. It could be anything, oxygen or carbon dioxide. So let me just draw a couple of air molecules here. So let's say that is a gaseous, just for the sake of argument, gaseous oxygen. So each of this is an O2, each of those, right? And let's say that this is the current configuration, that all of this is a vacuum here, and that there's some temperature. So these water molecules, they have some type of kinetic energy. They're moving in some type of random directions right there. So my question is, what is going to happen? What is going to happen in this type of container? Well, any of these guys are going to be randomly bumping into each other. They're more likely to bump into things in this down left direction than they are in the up right direction. So if this guy was happening to go in this down left direction, he's going to bump into something and then ricochet into the up right direction. But in the up right direction, there's nothing to bounce into. So in general, everything is moving in random directions, but you're more likely to be able to move in the rightward direction. When you go to the left, you're more likely to bump into something. So it's almost common sense. Over time, if you just let this system come to some type of equilibrium, and I'm not going to go into detail on what that means. You can watch the thermodynamics videos if you'd like to see that. You'll eventually see the container will look something like this. I can't guarantee it. There's some probability it would actually stay like this, but very likely that those 5 particles are going to get relatively spread out. This is diffusion. And so it's really just the spreading of particles or molecules from high concentration to low concentration areas. Right? In this case, the molecules are going to spread in that direction, from a high concentration to low concentration area. Now you're saying, Sal, what is concentration? And there's many ways to measure concentration, and you can go into molarity and molality and all of that. But the very simple idea is how much of that particle do you have per unit space. So here you have a lot of those particles per unit space, and here you have very few of those particles per unit space. So this is a high concentration, and that's a low concentration. So you can imagine other experiments like this. You could imagine a solution like, let's do something like this. Let's say I have two containers. Let's say two containers. Let's go back to the solution situation. So this was a gas, but I started off with that example. So let's stay with that example. Let's say that I have a door right there that's larger than either the water or the sugar molecules. On either side, I have a bunch of water molecules. I have a bunch of water molecules on either side, just like that. On either side. So I have a lot of water molecules. So if I just had water molecules here, they're all bouncing around in random directions. And so the odds of a water molecule going this way equivalent to odds of a water molecule going that way, assuming that both sides have the same level of water molecule. Otherwise, the pressures would be different. But let's say that the top of this is the same as the top of this. So there's no more pressure going in one direction or another. So if for whatever reason a bunch more water molecules were going in the rightward direction, then all of a sudden this would fill up with more water, and we know that isn't likely to occur. So this is, this is just a solution with, or this is just a kind of two containers of water. Now let's put some solute in it. Let's dissolve some solute in it. And let's say we do all the dissolving on the left-hand side. So we put some sugar molecules on the left-hand side. And these are small enough to fit through this little pipe. That's one assumption that I'm making. So what's going to happen? All of these things have some type of kinetic energy. They're all bouncing around. Well, over time, the water is going back and forth. This water molecule might go that way, that water molecule might go that way, but they net each other out. But over time, one of these big sugar molecules will be going in just the right direction to go through. Maybe this guy, instead of going in that direction, he starts off going in that direction. He goes just through this tunnel connecting the two containers, and he'll end up there, right? And this guy will still be bouncing around. There's some probability he goes back, but there's still more particles, more sugar particles here than there. So still there's more probability that so this guy's on that side, so let me cross him out. But there's still more probability that one of these guys will go to that side than one of these guys will go to that side. So you can imagine if you're doing this with gazillions of particles, I'm only doing it with 4, over time, the particles will have spread out so that their concentrations are roughly equal. So that, maybe you'll have 2 here over time. But if you're, when you're only dealing with 3 or 4 or 5 particles, there's some probability it doesn't happen. But when you're doing it a gazillion and they're super small, it's a very, very, very high likelihood. But anyway, this whole process, we went from a container of high concentration to a container of low concentration, and the particles would have spread from the low concentration container to the high concentration container. So they diffused. This is diffusion. This is diffusion. And just so that we learn some other words that tend to be used with the idea of diffusion, when we started off, this had a higher concentration. The left-hand side container had higher concentration. Higher concentration. Higher concentration. It's all relative, right? It's only higher than this guy. Higher concentration. And this right here had a lower concentration. Lower concentration. And there are words for these things. This solution with a high concentration is called a hypertonic solution. Let me write that in yellow. Hypertonic solution. Hyper, in general, meaning having a lot of something, having too much of something. And this lower concentration is hypotonic solution, lower concentration. You might have heard maybe one of your relatives, if they haven't had a meal in a while, say, "I'm hypoglycemic." That means that they're feeling lightheaded. There's not enough sugar in their bloodstream, and they want to pass out, so they want a meal. If you just had a candy bar, maybe you're hyperglycemic, or maybe you're just hyper in general. But so these are just good prefixes to know. But hypertonic, you have a lot of the solute, so you have high concentration. And then in hypotonic, not too much of the solute, so you have a low concentration. These are good words to know. So in general, diffusion, if there's no barriers to the diffusion, like we had here, you will have the solute go from a high concentration or hypertonic solution, if they can travel, to a hypotonic solution, to a hypo where the concentration is lower. Now, let's do an interesting experiment here. We've talked about diffusion, and so far we've been talking about the diffusion of the solute, right? And in general, and this is not always the case, if you want to be as general as possible, the solute is whatever you have less of. The solvent is whatever you have more of. And the most common solvent tends to be water. But it doesn't have to be water. It could be some type of alcohol. It could be it could be mercury. It could be a whole set of molecules. But water in most biological or chemical systems tends to be the most typical solvent. It's what other things are dissolved into. But what happens if we have a tunnel where the solute is too big to travel, but water is small enough to travel? So let's think about that situation. Let's think about that situation. In order to think about it, I'm going to do something interesting. Let's say we have a container here. Let's say we have, actually, I won't even draw a container. Let's just say we have an outside environment that has a bunch of water. This is the outside environment. And then you have some type of membrane. You have some type of membrane here. That's a membrane. Water can go in and out of this membrane. So it's semi-permeable. Well, it's permeable to water, but the solute cannot go through the membrane. So let's say that the solute is sugar. So we have water on the outside and also and also inside the membrane. So these are little small water molecules. This is a membrane right here. And let's say that we have some sugar molecules again. I'm just picking on sugar. It could have been anything. So we have some sugar molecules here that are just a little bit bigger, or they could be a lot bigger. Actually, they're a lot bigger than water molecules. So you have a bunch of, and I only draw 4, but you have a gazillion of them, right? But you have that much more water molecules. I'm just trying to show you have more water molecules than sugar molecules. And this membrane is semi-permeable. Semi-permeable. Permeable means that it allows things to pass. Semi-permeable means it's not completely permeable. So semi-permeable in this context, I'm saying I allow water to pass through the membrane. So water can pass. Water can pass. But sugar cannot. Sugar is too large. Sugar cannot. And the reason why in this case is because the sugar molecule is too big. So if we were to zoom in on the actual membrane itself, maybe the membrane looks like this. I'm going to zoom in on this membrane. So I'm going to zoom in on this membrane. So it has little holes in the membrane, just like that. And maybe the water molecules are about that size, so they can go through those holes. So the water molecules can go back and forth through the holes, just like that. But the sugar molecules are about that big, so they cannot go through that hole. They're too big for this opening right here to go back and forth between them. Now, what do you think is going to happen in this situation? So first of all, let's use our terminology. Remember, sugar is our solute, water is our solvent, semi-permeable membrane. Which side of the membrane has a higher or lower concentration of solute? Well, the inside does. The inside is hypertonic. Hypertonic. So I could write here, hypertonic. The outside has a lower concentration, so it's hypotonic. It's hypotonic out here. Lower concentration of solute. Now, if these openings were big enough, based on what we just talked about, these guys are bouncing around. Water is traveling in either direction and equal probability. Or I'm gonna talk about that in a second. If everything was wide open, it would be equal probability. But if it was wide open, these guys eventually would bounce their ways over to this side, and you would probably end up with equal concentrations eventually. And so you would have your traditional diffusion, where high concentration of solute to low concentration of solute. But in this case, these guys, they can't fit through the hole. Only water can go back and forth. If these guys were not here, water would have an equal likelihood of going in this direction as they would be going in that direction. A completely equal likelihood. But because these guys are on the right-hand side of, or in this case, on the inside of our membrane, this is our inside of our membrane zoomed up, these guys, it's less likely, because these guys might be in the kind of the approach position of the holes, it's that slightly less likely for water to be in the approach position for the holes. So it's actually more probable that water could enter than water exit. And I want to make that very clear. If these sugar molecules were not here, obviously it's equally likely for water to go in either directions. Now that these sugar molecules are there, these sugar molecules might be on the right-hand side. They might be blocking, I guess the best way to think about it is blocking the approach to the hole. They'll never be able to go through the hole themselves, and they might not even be blocking the hole, but they're going in some random direction. So if a water molecule was approaching, it's all probabilistic, and we're dealing with gazillions of molecules. It's that much more likely to be blocked to get outside. But the water molecules from the outside, there's nothing blocking them to get in. So you're going to have a flow of water inside. So in this situation, with a semi-permeable membrane, you're going to have water. You're going to have a net inward flow of water. And so this is kind of interesting. We have the solvent flowing from a hypotonic situation to a hypertonic solution. But it's only hypotonic in the solute. That's when you talk about the solute. And it's only hypertonic when you talk about the solute. But water, if you flip it the other way, if you view sugar as the solvent, then you could say we're going from a high concentration of water to a low concentration of water. I don't want to confuse you too much. This is what tends to confuse people. But just think about what's going to happen. No matter what, in what situation, the solution is going to do what it can to try to equilibrate the concentration, to make the concentrations on both sides as close as possible. And it's not just some magic. It's not like the solution knows. It's all based on probabilities and these things bumping around. But in this situation,
Okay.
water
He kind of finally got to it there close to the end. Was basically, he's talking about this, but what's going to happen is the water's going to move across the membrane because the solute can't, and it's going to try to balance out the concentration on the two sides of the membrane. Is what the membranes in the body tries to do, and that's the essence of osmosis. So it's the movement of water molecules instead of the solutes. So we can move the water molecules across the membrane when we can't always move the solutes, and then we can use that to try to balance the concentration of the fluids on the inside and the outside of the cell, which is kind of what Saul was trying to get to. Saul is like a genius. So he can very quickly talk over our heads, and he's comprehending things at a level that I don't even comprehend. So that's why he was talking a little bit different. But I wanted you to see that so you could kind of see a little more visual about what I was talking about. So if we keep looking at osmosis, he talked about hypertonic and hypotonic solutions. Basically, if you have a hypertonic solution, it has a higher concentrations of a given solute. Hypotonic solution has a lower concentration, and those are only relevant when we're comparing the two one to each other, right? So if we're comparing, say, a glass of sugared tea with a glass of non-sugared tea, then we would say that the glass of tea with sugar in it has a hypertonic solution, and the glass without sugar in it is a hypotonic solution. If we added half the amount of sugar that was in the sugared tea to the unsugared tea, the original sugared glass of tea would still be hypertonic in relation to the glass that we just added a little bit of sugar to, and that would still be hypotonic. So hypertonic, hypotonic, and even isotonic, which I'll talk about in a minute, are only relevant to when we're comparing two particular concentrations. So inside the cell versus outside the cell, when we're talking about hypertonic and hypotonic, that's relevant. Isotonic actually is exactly the same. So if I'm drinking pre-made sweet tea at the restaurant and the waitress comes around and refills my glass with the already sweetened tea, I'm not going to notice any difference because the solution of tea that she used to fill my glass would be isotonic. It would be exactly the same as what was already in there. So there's nothing that's going to change. There's no solution changes. There's no osmosis per se that's going on. I know it's not the perfect example, but hopefully you're getting my meaning. Isotonic, hypertonic, hypotonic are the solutions. And this one here, he talks about the different types of solutions. And hopefully that will help clarify things a little bit. I know he muddied the waters a little bit in that last one. Hopefully this one will clarify it.
So I have three different scenarios here of a cell being immersed in a solution. And the cell is this magenta circle, that's the cellular membrane. And then I have the water molecules depicted by these blue circles. And then I have the solute inside of the solution, inside of the water solution, that we depict with these yellow circles. And I've clearly exaggerated the size of the water molecules and the solute particles relative to the size of the cell, but I did that so that we can visualize what's actually going on. Now we're going to assume that the cellular membrane, this phospholipid bilayer, is semi-permeable, that it will allow water molecules to pass in and out. So a water molecule could go from the inside to the outside or from the outside to the inside, but we're going to assume that it does not allow the passage of the solute particles. So that's why it's semi-permeable. It's permeable to certain things, or we could say selectively permeable. Now, what do we think is going to happen? Well, the first thing that you might observe is we have a lower concentration of solute on the outside than we have on the inside. So in any given moment of time, you will have some water molecules moving in just the right direction to go from the outside to the inside. And you will also have some water molecules that might be in just the right place to go from the inside to the outside. But what's more likely to happen, and what's going to happen more over a certain period of time? Well, the water molecules that are on the outside, and we talk about this in the osmosis video, they're going to be less obstructed by solute particles. If this one happens to be moving in that direction, well, it's going to make its way to the membrane and then maybe get through the membrane. While something, if this water molecule was moving in this direction, well, gee, it's going to be obstructed now. Or maybe this is bouncing back and it's going to ricochet off of it. So the water molecules on the inside are more obstructed. They're less likely to be able to fully interact with the membrane or move in the right direction. They're being obstructed by these solute particles. So even though you're going to have water molecules going back and forth, in a given period of time, you have a higher probability of more going in than going out. And so you're going to have a net inflow.
And also realize this is going to be happening at much faster rate than what he's depicting.
Where we're talking about a cell and it's in a solution that has a lower concentration of solute. It's important that we're talking about a solute that is not permeable, that is not allowed to go through the membrane. The membrane is not permeable to that solute. We call this type of situation, this type of solution that the cell is immersed in, we call this a hypotonic solution. Hypotonic solution. And anytime we're talking about hypotonic, or as we'll see, isotonic and hypertonic, we're talking about relative concentrations of solute that cannot get through some type of a membrane. And the word hypo, you might have seen it in other things. It's a prefix that means less of something. So in this case, we have
Okay, hang on just a second. Let me pause him for a minute. You know, he's talking about this solution that's on the outside of that cell being a hypotonic solution. And I think it's important to point out when we talk about hypo, hyper, and isotonic solutions, we're talking about the solution outside of the cell and what that solution is relative to what's inside the cell. So when he's talking about it here, it's a hypotonic solution that's outside of the cell because the concentration is much lower outside the cell than inside the cell. So it's hypotonic relative to what's going on inside the cell, okay?
Concentration of solute in the solution that we have inside of the cell, and because of that, you're going to have osmosis. You're going to have water molecules going from the inside, or you're going to have water molecules going from the outside, I should say, to the inside, and that's actually going to put pressure on the cell. The cell itself might expand, or it could even, if there's enough pressure, it might even explode. So now let's go to the next scenario. In this scenario, we have roughly equal concentrations of solute on the outside and on the inside. At least I tried to draw them that way. So in this situation, the probability of a water molecule in a given period of time going from the outside to the inside or from the inside to the outside is going to be the same. And so you're not going to have any net inflow or net outflow. You're always going to have water molecules going back and forth, but there's not going to be any net inflow or outflow. And so in this situation, so let's see, let me write no net flow. And this type of solution, where you have the same concentration of solute in the solution as you do inside the cell, we would call this an isotonic. This is an isotonic solution. Isotonic solution. And the prefix iso refers to things that are the same. It has the same concentration of solute. And so you have no net inflow. So hypotonic solution, you have water molecules going into the cell expanding, kind of like a filling balloon. Isotonic solution, no net flow. And then of course, you can imagine in this last scenario, I have a higher concentration of solute on the outside than I have on the inside. And we can guess what's going to happen. So first, what would I call this? Well, I have more of something in the solution, so I would use the prefix hyper. I have more of it, more hypertonic. This is a hypertonic solution. And once again, the solute can't go across the membrane, but the water molecules can. And you're going to have water molecules going from the outside to the inside, and from the inside to the outside. But the probability that the ones going from the ones on the inside are going to be less obstructed to go out than the ones on the outside to go in. And so you're going to have a net outflow. You have a higher probability of things going from the inside to the outside than you do from things going from the outside to the inside, because they're going to be more obstructed. And so they're going to be held back, I guess, in different ways. So in this situation, you're going to have the water escape the cell, and the cell actually might shrivel up. The cell, since it's going to lose that pressure from the water, the cell itself might shrivel up in some way. And you can actually see this in actual living systems. If you were to put a red blood cell into a hypotonic solution, the water is going to rush into it, and it's going to blow up. It's going to expand. And so it's going to look like a overinflated red blood cell. In an isotonic solution, it's going to look the way that we're used to seeing a red blood cell, actually having kind of that little divot in the middle area, while over here it's all going to expand. And then in the hypertonic solution, the water is going to escape the red blood cell, and then you would actually see it kind of shrivel up a little bit like this, because all of that water is we have a net outflow of water molecules.
Okay. Oh, sorry, I didn't want to go there. Let me back up. There it is. There where I'm going to try to, just bear with me for a second. I'm going to try to get this back to where we were down here at the end and pause it. Nope, pause. Pause. There we go. So he's talking about hyper, hypo, and iso. Why is that important? Why is it important that you know that? One of the things that we routinely give to patients in the hospital, and even outside of the hospital, me as a paramedic, is something called normal saline. Okay, normal saline. Why is it called normal saline? Well, it contains 0.09% sodium chloride. Okay. 0.09% sodium chloride. That just so happens to be the same sodium chloride concentration inside of our bodies. So it's the same. That means if we give somebody normal saline, it's going to be considered an isotonic solution. So normal saline is an isotonic solution. We give somebody normal saline, it's not going to cause the water to move in and out of the cell at all. Okay? It's not going to cause any of that stuff to happen. Is there ever a time when we give somebody a hypertonic solution, say, 0.18 normal saline? Well, I'm not going to say there's not. I'm sorry, I put normal saline. There's nothing normal about 0.18. Let me erase that, fix that for us. So 0.18 NaCl. So 0.18%, that's a percentage, sorry. percentage of sodium chloride. So is there ever a time when we would have that? No, I mean, not really. It's unlikely that you're going to be using something like that. It's not impossible though, but it's unlikely. I will tell you something that we do see, and let me get rid of this NS up here. It's kind of in my way. Something that we do see is 0.45% sodium chloride. That's something that we do commonly see, the purpose of that, actually, we see that used not all the time, but we see it used sometimes with diabetics that have a really, really, really high blood sugar. They haven't taken their insulin and done the things that they need to do. They have a really high blood glucose level. Glucose can be very dangerous in high concentrations inside of cells. So what we could do is we could give them half normal saline, and that would change it makes it a hypotonic solution. So we would wind up with what's happening here is we would wind up with more water going into the cell and basically diluting down the concentration of glucose that's inside of that cell. So that's kind of what happens, and that's why knowing the osmolarity of cellular solutions, or the solutions that we're putting into somebody's body, is important. Most of the solutions that we give people are isotonic. They're not going to cause fluid shifts from inside of the cell or to outside of the cell, and there's reasons for that. But if we do happen to give a solution that's going to pull fluid out of a cell or push fluid into a cell, there's a doggone good reason for doing that. Not uncommon with the half normal saline. This is what we refer to as, this one up here, we often call it half normal saline because it's half of the normal. No, we don't call this one over here double normal or anything like that. Not that I know of. I can't think of an instance where double the concentration of normal saline or a higher concentration of sodium chloride would be used. But I'm sure there probably is. It's just not something that's very commonly done, and that's why I'm unfamiliar with it. So hopefully you got a better understanding of hypotonic, isotonic, and hypertonic solutions. Here's those red blood cells he was trying to show us a picture of. Thought he did a really good job of showing that, of what those cells are going to look like given in different solutions. Same picture in your book. So there is, when we continue on with the types of diffusion, there's facilitated diffusion. So those lipid insoluble things in large substances have to be transported by facilitated diffusion. Glucose is one of those. It's too big. And basically we've got these membrane channels or protein channels that act as carriers to bring that stuff in and out or through that cell membrane. It's facilitated, so it takes a little bit of work to get it to go through there. There are carrier mediated and channel mediated. Some of these are as simple as opening up a channel. You know, a trigger opens up a channel, and this protein just opens up, and things just flow through it. Certain substances just flow through them. But there are some that are actually triggered by the presence of something that causes the protein to change shape to where that can cross in there. Those are not necessarily, these are not, and I may have mentioned that they were. These are still passive. These are not taking up energy to do it. It's just this protein changing shape in the presence of that particular substance. But it requires that protein in order for it to get in and out of the cell, so it can't move across there very fast. So that's kind of the issue with, say, glucose. It can't move across there very fast, so we have to have a means to get it across there. Water and solutes can be forced through a membrane by fluid or pressure that we put on it. So pressures play a role in how water and solutes can be moved through a membrane. So what we do when we have a solute concentration of a higher concentration and a lower concentration, Saul didn't really mention this, but what happens is we do kind of create a pressure gradient. There's more pressure on one side. The higher the solute concentration, the higher the pressure on that one side. This is going to cause things to kind of push across to the other side and to try to balance them out. It's a little bit more complicated than that, but it is a process. It's part of the filtration process that actually happens in the kidneys. But pressure can play a role in that. Pressure plays a small role in that when we're talking about the hypotonic things. Not as much. It is a lot about just the kinetic energy of those things bouncing around. But pressure can play a small role in that. And Saul was just trying to keep from confusing things too much, like I probably just did. There is an active process, sometimes called solute pumping. It's where there's a protein that requires that protein carriers that transport those substances. Those that are just too big to travel through the membrane, maybe not lipid soluble, and may have to move against the concentration gradient, which the cells do that. Cells will intentionally have substances move into or out of the cell against the concentration that's on the inside or the outside of the cell. And to do that, it's an active process and it requires a pump. ATP is used for that process because it's active and it takes work, but it's through the use of proteins, but it's not channel proteins. It's pumping proteins that actually pump that stuff out. Let's see, and this gives us an example of some amino acids and some sugars and some other ions that are pumped in and out, and ATP is being used. A good example of this is the sodium-potassium pump. Sodium-potassium pump is used in many cells in the body, and especially those cells that have what we call excitability. So heart cells, muscle cells, nerve cells all rely on the sodium-potassium pump to pump sodium out of the cell, and I may have this backwards. Yeah, oh yeah, there it is right there. Duh. Sodium out of the cell and potassium into the cell, regardless of the concentration differences. So it'll intentionally pump more sodium out of the cell to create a solute inside of the cell that has less sodium in it. And it'll pump more potassium into the cell to have a higher concentration of potassium. So those special cells, what they'll do is they'll intentionally have more sodium outside of the cell and more potassium inside of the cell. And that gradient that it creates is for a purpose. It actually uses that gradient to its benefit so that it makes the membrane able, or it makes that cell able to perform a task or to perform a thing. And which we'll talk about later how that process works, that sodium-potassium pump process and what it's for, and membrane excitability. But just realize that those pumps are actually going to pump sodium out and potassium in, and they're going to create concentration differences on the inside and outside of the cell intentionally, and that's what creates a very sensitive membrane on that particular cell, to where that cell can do a task like contract like muscles do, or contract like the heart cells do, that kind of thing. So that's what we're looking at. This is a little video about sodium-potassium pumps and what they do. We'll just watch it.
What I hope to do in this video is give ourselves an appreciation for the sodium-potassium pump. And as the name implies, it pumps sodium and potassium, but it does it in different directions. So this little depiction right over here, this is my drawing, my rendition of the sodium-potassium pump. It's a transmembrane, I guess you could say protein complex right over here. And in this resting state, it is open to the inside of the cell, and it has an affinity for sodium ions. And so the sodium ions, you see 3 sodium ions depicted here in blue. They're going to bind to the pump, and once they bind to it, then it's going to want to be phosphorylated by an ATP, and we see that right over here. This is ATP, adenosine triphosphate, and when it gets phosphorylated, it's a release of energy, and it allows the conformation of the actual protein to change. So the new conformation of the protein, it's now going to open up to the outside, close off to the inside, and now it's no longer going to have an affinity for sodium ions, but an affinity for potassium ions. And this is fascinating, that release of energy, change of conformation, that these proteins really are these molecular machines, these fascinating molecular machines. But once that happens, this change of conformation, the sodium ions are going to be released outside of the cell, and then you're going to have potassium ions that are going to bind from the outside. And then once that happens, the change in conformation, we're going to have it's going to get dephosphorylated, and then you're going to go back to your original conformation right over here, where you no longer have an affinity for potassium ions. They're going to be released, and then you're going to be back in the original phase. So this is fascinating. By using ATP, by using energy, this is active transport. It takes energy to do this. Let me write this down. This is active transport that we are talking about right over here. We're able to pump, using an ATP, we're able to pump 3 sodium ions out. 3 sodium ions out. So let me write that down. 3 sodium ions out, and in the process, we pump 2 potassium ions in. So we pump two potassium ions in. Now you might say, okay, the outside, since these are both have positive charge, but I have 3 sodium going out, 2 potassium going in, that must make the outside more positive than the inside, and that actually is true. But that by itself isn't fully responsible. It's actually only partially responsible for the electric potential difference between the inside of the membrane and the outside of the membrane. What really sets that up is that you actually have channel proteins that allow potassium ions to move down, to diffuse down their concentration gradient. So let's think about what happens. Before I even talk about these channel proteins right over here, because of the sodium-potassium pump, what is sodium's concentration gradient? Well, it has a higher concentration on the outside, and it has a lower concentration on the inside. This is sodium's concentration gradient. What is potassium's concentration gradient? Well, potassium is getting pumped in from the outside into the cell. So potassium has the opposite concentration gradient. It has a high concentration inside, and it has a low concentration outside. Now, if we let potassium go through, we've talked in previous videos about ions just not being that permeable, that just the general membrane, if it's not facilitated in some way, isn't that permeable to things like ions, like sodium and potassium ions. But if you have channel proteins right over here that let the potassium get out, what's going to happen? Well, you might have one of 2 answers. You might say, well, look, hey, things diffuse down their concentration gradient. You have a higher probability, since you have more potassium here than up here, higher probability of them going in the right direction on this side and moving from this side to that side than you have them going from that side to that side. And so you would have a net outflow of potassium. And some of you might say, well, okay, that makes sense if you only care about the concentration gradient. But what happens if we look at the charge? Because we're saying that the inside of the cell is going to be less positive, and the outside of the cell is going to be more positive because it has more, that we have the net ion change. So it's going to be more positive out here. So positive ions, you want to move away from charges that are the same as you. You want to move to the places that are more negative. So you'd say, well, these potassium ions are positively charged. Why would they want to go from a less positive place to a more positive place? And if you are saying either one of these things, talking about the concentration gradient or talking about the electric potential difference, you are actually going to be right in both cases. These are going to be balancing forces. The concentration gradient is going to allow some of these potassium ions to pour out, but the concentrations of potassium ions aren't going to fully equalize because of the electric potential difference. Because, hey, it's more positive out here, it's less positive here. When they're moving out, they're going against what their charge wants to do. They're going with the concentration gradient, but at some point that is going to balance out. And by going through this process, by pumping sodium out and with a larger ratio than what you're pumping potassium in, and then you're further allowing more positive charge to go out, you're establishing what's called the resting membrane potential for a cell. And this is super important for all cells, but especially neuron cells or neural cells or neurons. And those are going to spend 2/3 of their energy just to establish or to keep the resting membrane potential. And as we'll see in the videos on neurons, that's because they keep leveraging that potential to send signals down the neuron. But the resting membrane potential, it's less positive here and more positive there. If you measure this relative to, let me make that a little bit neater, relative to this right over here, this difference is, depending on what estimates you look at, approximately negative 70 millivolts. I've seen estimates negative 60, negative 80, negative 70 millivolts. millivolts. And this is key for neurons, but it's key for all cells. Now, the sodium potassium pump isn't just about establishing.
Okay, so we kind of get, at least I hope you get, the purpose and the function of the sodium potassium pump. Basically, we're pumping 3 sodiums out, 2 potassiums in, and because we're pumping more sodiums out than we are potassium in, we create that negative, that voltage difference between the inside and the outside of the cell. And over here where Saul said it created about a difference of about negative 70 millivolts. And that's in that membrane, in the potential of that membrane because of the voltage difference between the two. Now, if we stimulate that membrane with something, channels will open up and sodium and potassium is going to rush in and out, or it's going to do different things like that. And it's going to cause that cell to react. So that's why it's important that the sodium and potassium pumps work properly in order for that cell to be primed and ready to do a specific function, to do a certain function. And we call that a cell's resting potential once it reaches that point. But realize those sodium potassium pumps are going nearly all the time, but maybe not at a fever pitch until we stimulate that membrane and it's allowed a whole bunch of sodium to rush in the cell and potassium to rush out of the cell because we opened up sodium and potassium channels when we stimulate the membrane. And when we do that, those sodium and potassium pumps have got to work overtime to get everything back to normal. But also they're going to be working a little bit the whole time anyway, just not real crazy, because of those leak channels where we're leaking some potassium out, or maybe or sodium may be leaking back in through the membrane. So that's kind of how that sodium and potassium pump works. We'll talk more about sodium and potassium pumps later. We'll talk more about membrane potentials and those types of things later. So it's not the last time that you're going to hear about those things. But you may want to, at a later date, go back and look at those videos. They'll probably make more sense a little bit later. And now you may be saying, well, if they'll make more sense later, why in the world are you making me watch them now? Well, because they're still relevant now. And even though you maybe don't understand all of it, you understand enough of it to where that we can move on. You need to have some sort of understanding so that we can move on, and we'll still build on that a little bit later in some other chapters. So, another form of the active transport process is vesicular transport. And that's basically moving substances in and out or across a plasma membrane through 2 means, excitosis or endocytosis. Basically, excitosis is where we have a substance that's inside a vesicle inside of that cell. We take it out to the edge of the cell membrane and we basically set it free. Endocytosis is where the cell actually grabs a hold of something, that membrane kind of drinks it in and brings it in as a vesicle. I think there's a couple of pictures here. And that's kind of excitosis that talks about that just a little bit. But the best example is this down here on the bottom. I know we've seen this picture before, but this is an example right here of this coming out is excitosis. So this vesicle here was created by the Golgi apparatus. It floats through the inside of that cell, comes up on the membrane, contacts with the cell membrane, lets its contents out, and this vesicle is just absorbed by the membrane. The reverse of that would be, and maybe I'm hoping there's another slide maybe that clarifies that. Yeah, endocytosis is a little different. Here's no, that's excitosis, sorry. That's actually an electron microscope picture of excitosis of a vesicle getting to the edge of a membrane. Endocytosis is a little bit different. It's the opposite, and this is really wordy where it's talking about this, but basically you can see here where this cell has created a little bit of a pit. It takes in these little molecules here, whatever it is, it ingests, pinches off, moves, well, I don't know how it did that, but moves across into here. It's utilized or whatever, and whatever's needed and whatever, and then whatever is not needed from the contents of that are dumped back off on the other side through excitosis. That's just endocytosis. It's just a way that the cells can take in components that can't readily move through the membrane, or that may be too dangerous to be freely loose inside of the membrane. So that's all that is. And that's just going to go through one at a time. Oops, let me back up. There's also another type of endocytosis is phagocytosis. It's cellular eating. It's basically where one immune cell grabs a hold of another cell and devours it. Bacteria, sometimes dead body cells, that kind of thing. These phagocytes will go around, and they're a protective mechanism in order to protect the body and rid it of things that don't belong. So that's basically what it's doing. Okay. And then there's the penocytosis is cellular drinking. It's where a cell can actually, gulp droplets of extracellular fluid that have proteins or fats, that kind of thing. For whatever reason, that cell may be growing, maybe enlarging for whatever reason. It needs to take in some more of those fluids in order to do what it does. Cells routinely do this, and especially those that are involved in an absorption, such as the small intestine. It's not unusual for them to drink extracellular fluid. Similar deal where it creates that pit, drinks in the stuff that it needs and releases it inside. There's different types of endocytosis, such as the receptor-mediated endocytosis. It's used to take up specific molecules that are needed, and then there's receptor proteins on the membrane surface that bind to that substance. So we have a protein on the outside of the membrane, something that comes by that the cell needs. That protein latches onto it, holds onto it. It's pitted in and it's brought into the cell and then utilized for creating something, usually such as enzymes or hormones. Sometimes cholesterol and iron are ways of are brought in that way. And then, of course, both the receptor and the target molecule are brought into that vesicle. And usually the target molecule or the receptor is replaced. It's brought back out to the surface of it. Kind of like we see here. It's exactly that same picture that they're showing us over and over again. But we see this. Here's a good example of it in this picture. It shows these right here are the proteins that are specific, meaning these are the only things that can plug into there. Once enough of them have plugged into it, it will pit out and pinch off, take that into the other side, or take that into the inside of the cell, dump that off, and then on the other side probably pick those proteins back up to where that they can be reused. Each cell goes through a life cycle, a series of changes from the time it's formed until it divides. So there is a cellular life cycle. There's two major periods of the cellular life cycle when cells are growing and dividing and all that stuff. There's the interphase, which has to do with cells growing, carrying on the metabolic processes and all that stuff. It's the longer phase of the cell cycle. And then, of course, there's cellular division, is where that cell replicates itself. Not all cells are capable of replicating themselves. Only certain cells are capable of doing that. And once, then there's some cells that only do that for a period of time, and then they stop doing that, and they're unable to continue to do that. One of the better examples is bone cells, whereas the growth plates that you hear people talk about. Oh, don't break your leg on the growth plate because it'll make one leg shorter than the other. There's a lot of truth to that, because those particular cells, the stem cells that are creating more bone cells, once they get encased and entrapped in a bony matrix, they actually lose their ability to divide. So as long as they're still in that cartilage of that growth plate, they're still able to keep dividing and creating more bone cells. But once they get kind of walled in, it's over with. They're not gonna divide anymore. During that, the cellular division, genetic material is duplicated and it gets ready for cell division into two cells. It occurs towards the end of the interphase once everything is ready. Remember, DNA is very complex. It's going to take some time. that process needs to be done perfectly and it needs to be done correctly. Otherwise, we're going to wind up with a cell that can't function the way that it's supposed to function. If there's something that interferes with that process of the DNA replication, we can have real consequences, real problems. The envelope that the DNA resides in inside of that nucleus is pretty well protected. But one of the things that we can do that can greatly damage the DNA inside of that nucleus, inside that nuclear envelope, is go lay out in the sun and get a suntan. We expose that nuclear envelope to enough radiation. Eventually, some of that radiation leaks through, and it can damage this DNA. Maybe it damages it during the replication process, and then the cells that are created from that are rogue cells. They don't work properly. They're kind of do their own thing, and they can be damaging to the rest of the body, and we call them cancer cells. That's one of the ways that if we damage the DNA improperly. Otherwise, the worst extreme of that is to damage the DNA to the point that it just kills it, that it can no longer function, which is the purpose actually of radiation therapy. When we're trying to treat cancer, we're trying to just kill off its DNA so it can't function anymore. Kind of ironic. So during that replication of DNA, it's going to uncoil into two chains. Each side serves as a template. And then the adenine, guanine, and thymine, and cytosine, and all those stuff are going to bond together. It's going to take in all of those to where that DNA is going to split into two separate chains, or two halves of a ladder. These complementary components are going to plug onto that, and actually they'll plug on there with the help of some proteins. And then what will happen is we'll wind up with two complete copies of that DNA. So we see here where it kind of splits apart here, and then two chains are created. And I think this is pretty easy to understand, but it splits apart right here, and then these begin to attach onto it, and that creates two completely separate chains of that DNA, which is that part of that DNA replication process. And then, of course, mitosis is the division of the nucleus. So that's when the nucleus divides into two daughter nuclei. And then cytokinesis is the division of the cytoplasm, which is kind of towards the end of it. It begins when mitosis is almost done, and it results in the two daughter cells actually being created from the one. So that's cellular division in a nutshell. Mitosis actually has some phases of its own. So the division of the nucleus kind of goes through its own set of phases. It's got the prophase where the chromatin coils into chromosomes. You know, we talked a little bit about chromosomes. Please don't try to get too crazy about understanding this. Mainly what I want you to understand a little bit about is mitosis, cytokinesis. Don't worry about the details of those two, such as the prophase and all of that. But this kind of gives you an idea of how that whole thing, you know, the centrioles and all that stuff begin to create the microtubules and the structures within the cell, and the nuclear envelope breaks them down and all that stuff. So, and then it goes into the metaphase, and then the anaphase and the telophase, and then we get cytokinesis where the cells actually divide into two. Whoops. This little video, and I really like these videos a lot better than poor Saul. Saul is so kind of dry and just to the point, and so he's such a brainiac. Sometimes for me, I like listening to him. I like that stuff, but he's not everybody's cup of tea. These are some of the crash course videos. Let these guys talk about mitosis. Or this guy here, he's fun to listen to.
Hey, check this out. Cool, huh? I bet you wish you could do this. Have a clone, clean up around the apartment for you, go to class, maybe take your mom out to dinner on her birthday. Well, you can't do that, and actually there are some really good reasons why you can't do that. We're going to talk about those in the next episode. But you know what can clone themselves? your cells, like almost every single one of them. And in fact, they're doing it right now. For any creature bigger than a single-celled organism, all of life stems from cells' ability to reproduce themselves, because that's what allows organisms to develop and grow and heal and keep from dying for as long as possible. This particular kind of cell division is called mitosis, and it's responsible for a whole lot of your body's key functions. If you get a cut, your body needs to make new cells, mitosis. Have too much to drink, damage your liver, you gotta replace those cells, mitosis. Tumor growing in your spine? Unfortunately, again, mitosis. While you go from a 7-pound baby to a 70-pound child, it's not your cells that are increasing in mass, you're just getting more of them over and over and over again. That's mitosis. This process is so central to your life that it will take place in your body over your lifetime about 10 quadrillion times. That's 10,000 billion times. Like all split ups, it's not easy. Gonna maybe be a little bit messy. There's a lot of drama, and it can take a surprisingly long amount of time, but trust me, after we're done with it, we'll all be better off. So you are made of trillions of cells, just like giraffes and redwood trees. And remember that inside each cell, there's a nucleus that stores your DNA, which contains all of the instructions on how to build you. That DNA is organized into chromosomes, and as we've mentioned before, in your body cells or somatic cells, you have 46 chromosomes grouped into 23 pairs. One in each pair is from your mom, and the other one's from your dad. Cells with all 46 chromosomes are called diploid cells because they have 2 sets each, and that's what we're focusing on today. You also have haploid cells that have half as many chromosomes, 23, and those are your sex cells. They're produced in an equally fantastic process called meiosis, which we'll be talking about in the next episode. But for now, the main thing to remember about mitosis is that it allows one cell with 46 chromosomes to split into 2 cells that are genetically identical, each with 46 chromosomes, all in order to keep the party of life going. Now, the nucleus in your cell controls everything that goes on in the cell. It has all of the instructions necessary for making the cell survive, so you don't need to duplicate the whole cell. All you need to do is duplicate the DNA, get it wrapped up, and then if you have 2 separate pockets of DNA, that's all you need to have 2 new cells. Mitosis takes place in a series of discrete stages called prophase, metaphase, anaphase, and telophase. And you could just say that over and over again and let it sink into your head. And part of what's, really amazing about this whole process is that while we know what these stages are, we don't always know the underlying mechanisms that make all of them happen. And this is part of science. Science isn't like all the stuff we know, it's how we're trying to figure all this stuff out. Consider it job security. If you ever want to be a biologist, there is a lot of stuff that future biologists have to still figure out. And this is one of them. All right, let's get our clone on. So most of their lives, cells hang out in this limbo period called interphase, which means they're in between episodes of mitosis, mostly growing and working and doing all the stuff that makes them useful to us. During interphase, the long strings of DNA are loosely coiled and messy, like that dust bunny of dog fur and laundry lint under your bed. That mess of DNA is called chromatin. But as the mitosis process begins to gear up, lots of things start happening in the cell to get ready for the big division. One of the more important things that happens is that this little set of protein cylinders next to the nucleus called the centrosome duplicates itself. We're going to have to move a lot of stuff around in the nucleus, and that's going to be regulated by these centrosomes. The other thing that happens is that all of the DNA begins to replicate itself too, giving the cell 2 copies of every strand of DNA. To brush up on how DNA replicates itself like this, check out this episode and then come on back. Now the cell enters the first phase, or the prophase, when that mess of chromatin condenses and coils up on itself to produce thick strands of DNA wrapped around proteins. Those, my friends, are your chromosomes. Instead of dust bunnies, the DNA is starting to look a little bit more like dreadlocks. And the duplicates that have been made don't just float around freely, they stay attached to the original, and together they look like little X's. These are called the chromatids, and one copy is the left leg and arm of the X, and the other copy is the right leg and arm. Where they meet in the middle is called the centromere. Just so you know, these X's are also called chromosomes, sometimes double chromosomes or double-stranded chromosomes, and when the chromatids separate, they're considered individual chromosomes too. Now, while the chromosomes are forming, the nuclear envelope gets out of the way by, completely disintegrating. And the centrosomes then peel away from the nucleus, start heading to the opposite ends of the cell. As they go, they leave behind a wide trail of protein ropes called microtubules, running from one centrosome to the other. You might recall from our anatomy of the animal cell that microtubules help provide a kind of structure to the cell. And this is exactly what they're doing here. Now we reach the metaphase, which literally means after phase, and it's the longest phase of mitosis. It can take up to 20 minutes. During the metaphase, the chromosomes attach to those ropey microtubules right in the middle at their centromeres. The chromosomes then begin to be moved around, and this seems to be being done by molecules called motor proteins. And while we don't know too much about how these motors work, we do know, for instance, that there are 2 of them on each side of the centromere. These are called centromere-associated protein E. So these motor proteins attached to the microtubule ropes basically serve to spool up the tubule slack. At the same time, another protein called dynein is pulling up the slack from other ends of the ropes near the cell membranes. After being pulled in this direction and that, the chromosomes line up right down the middle of the cell. And that brings us to the latest installment of biolography.
So how do chromosomes line up like that? We know that there are motor proteins involved, but like, how? What are they doing? Well, remember when I said earlier that there were a lot of things that we don't totally understand about mitosis? It's sort of weird that we don't, because we can literally watch mitosis happening under microscopes. But chromosome alignment is a good example of a small detail that has only very recently been figured out. And it was a revelation like 130 years in the making. Mitosis was first observed by a German biologist by the name of Walther Flemming, who in 1878 was studying the tissue of salamander gills and fins when he saw cells' nuclei split in 2 and migrate away from each other to form 2 new cells. He called this process mitosis, after the Greek word for thread because of the messy jumble of chromatin, a term he also coined that he saw in the nuclei. But Flemming didn't pick up on the implications of this discovery for genetics, which was still a young discipline. And over the next century, generations of scientists started piecing together the mitosis puzzle by determining the role of microtubules, say, or identifying motor proteins. Now, the most recent contribution to this research was made by a postdoctoral student named Tomomi Kiyomutsu at MIT. He watched the same process that Flemming watched and figured out how at least one of the motor proteins helps snap the chromosomes into line. He was studying a motor protein called dynein, which sits on the inside of the membrane. Think of the microtubules as tug-of-war ropes, with the chromosomes as the flag in the middle. What Kiyomutsu discovered was that the dynein plays tug-of-war with itself. Dynein grabs onto one end of the microtubules and pulls the tubules and chromosomes toward one end of the cell. When the ends of the microtubules come too close to the cell membrane, they release a chemical signal that punts the dynein to the other side of the cell. There, it grabs onto the other end of the microtubules and starts pulling until smack, it gets punted back again. All of this ensures that the chromosomes will line up exactly in the middle so that they will be split evenly. That discovery was published in February of 2012, a couple of weeks before I sat down in this chair, and 134 years after mitosis was first observed. If you want to join the ranks of scientists who are answering the many questions left about mitosis and lots of other things about our lives, maybe someday I'll do a biolography about you.
Now, so far we've gone through the interphase, where the centrosomes in DNA replicate themselves and get ready for the split. The prophase, where the chromosomes form and the centrosomes start to spread apart. And the metaphase, where the chromosomes align in the middle of the cell. And now, it's time to separate the chromosomes from their copies. This time, motor proteins start pulling so hard on the ropes that the X-shaped chromosomes split back into their individual single chromosomes. Once they're detached from each other, they're dragged toward either end of the cell. Since the prefix ana means back, that may help you remember the name of this phase, called anaphase. After this, it's just a matter of using all that genetic material to rebuild so that the copied genetic material has all of the accoutrements of home. In the last phase, telophase, each of the new cell structures are reconstructed. First, the nuclear membrane reforms, and the nucleoli form within them, and the chromosomes relax back into chromatin. Then a little crease forms between the 2 new cells, which marks the beginning of the final split. That division between the 2 new cells is called cleavage, and all that's left is to make a clean break. This is done by cytokinesis, literally cell movement, by which the 2 new nuclei move apart from each other and the cells separate. We now have 2 new cells, each with a full set of 46 chromosomes. These clones are called the daughter cells of the original cell, and like identical twins, they are genetic copies of each other, and also of their parent. But of course, that's obviously not the case for you. Even if you are an identical twin, shout out identical twins! See me in the comments. While you kind of are a clone of your sibling, you are not a clone of your parents. Instead, half of your DNA in each of your cells is from your mom and half is from your dad. To understand why that is, we have to understand how eggs and sperm are formed, and that is meiosis. And that's what we're going to be talking about next week on Crash Course. Until then, you can just watch this video over and over again, or you can just watch the bits that you want to re-watch using our table of contents, which is also available in the description for people who are using iPhones and can't click annotations. If you have any questions, you can reach us on Facebook or on Twitter.
All right, enough about cellular division, huh? I figured that was probably the easiest way to learn about that rather than me try to go through all that. And he's already talked about that. Another thing that DNA does is it is the blueprint for making protein. So it's responsible for protein synthesis. Basically, a segment of DNA carries that blueprint. And then, of course, those proteins, once created, are going to have a whole lot of different functions in the body. Just realize that there's a lot of proteins in the body, serve a lot of different purposes, and they're all vitally important, but they're all produced from DNA. Basically, DNA is the framework. It has these coded triplets that contain the three bases, which call for a particular amino acid that binds to them. Anyway, without getting too crazy about it, basically what happens is DNA never leaves the nucleus, but protein is actually created outside of the nucleus. So there's got to be a way to do this. Well, what happens is DNA unravels, much like very similar to the way that it does when it's splitting into two cells. It'll unravel and create a section of what's called RNA, which is a messenger form of DNA. The RNA then can leave the nucleus and actually it actually leaves the nucleus, and it goes into usually rough endoplasmic reticulum. There in the rough endoplasmic reticulum at a ribosomal site, it creates a protein. It's basically the message. It transcribes the information from the DNA, and it takes it out to the ribosomes in order to create those proteins. And that's going to happen in two phases, of course, transcription and translation, where the RNA is transcribed and then it's translated into the protein. And I don't really want us to get too in depth into that. Just want you to kind of have an idea of what's going on. So we can see here in this picture that the DNA has split apart and allowed for the creation of this strand of RNA right here. Okay, so it's creating this strand, and it's complementing, based upon along this line, and eventually this what's called RNA. This is going to be RNA when it's done, and it's called mRNA, stands for messenger. So it's messenger RNA, and it's going to come out of here. It's going to come out of the pore of the nuclear envelope. It's going to come out of there, and then it is going to, and of course that's part of the translation, the messenger RNA, and then it's going to get to one of these ribosomes. It's one of these large ribosomes, and these ribosomes are going to plug these little condoms on there and create proteins by plugging these on there in successive order until it's gone all the way down the length of this whole thing, this RNA, and created that particular protein based on these indicators, whatever you want to call it. So this is called tRNA. So it's because it's transposing or transponding, I can't think of the right word, but it's plugging into there and creating that protein. So that's essentially what happens with that. it's created and then it's translated, and the end result is it's created a protein. Feel free to watch this video. I've got this video on here, but feel free to watch it. I would play it, but doggone it, we're already into it for 2 hours. This video is also available under that same folder. Watch this video. It'll give you an idea of this transcription and translation of DNA and how it creates proteins. Okay? All right. Now, I'm going to whiz through these last slides pretty quickly. Body tissues are groups of cells with a specific structure and function. There's 4 different types of body tissues: epithelial tissues, connective tissues, muscles, and nerves. We're going to talk about all of these in different chapters. Some of them we'll talk about several different types in a given chapter. Some of them we'll spend a whole lot of time talking about that one particular one. And like this one here and this one here, we'll spend a lot of time talking about those. And we'll cover a lot of information on those. But those are the different types of tissues. Epithelial tissues cover the body, line the internal parts of the body like the digestive tract. Also some glandular tissues. They have these 4 functions: protection, absorption, filtration, and secretion. Don't panic over what I'm doing right now, and I'm whizzing through this. This is not stuff that's going to be on the test, because actually I believe it's chapter 4 we'll start talking about all of these things, these connective tissues and all that. If not chapter 4, it's chapter 5, and we'll talk about it. We'll rehash all that. So don't worry about knowing this stuff super well right now for the purpose of this book or this chapter. We're going to talk about it later. With epithelial cells, the characteristics of them is they fit really close together, and they often form sheets like layers in our skin, fit tightly together, water barrier, that kind of stuff. So when we look at that, we can see them here. Here's an example of those right there. Okay? Epithelia, their epithelia are actually classified by the number of layers. If it's simple, it has 1 layer. If it's stratified, it has more than 1. Okay, so we can see the difference: the top simple and the bottom stratified. Also, it's classified by the shape of the cells. There's squamous, cuboidal, and columnar. So different shapes of the cells. Also, the way that we classify it. There's no real crazy science to this. Squamous is kind of short and squishy. Cuboidal looks like a cube. Columnar looks like a column. So nothing really crazy about that. And this little chart here shows us kind of their purpose and what they do, the function that they serve. The reason why they have a different shape is because that's what is needed in that particular area. Remember, form follows function. Simple squamous, we find that wall and lining the walls of the capillaries, lining the sacs of the lungs. It's thin, it's squamous, it allows certain things to pass through it, which it needs to for those areas. Also serous membranes that line around organs and those types of things. It's where we'll primarily see those, just a real thin layer. Cuboidal, we see them in glands and ducts, the walls of the kidneys, surfaces of the ovaries. They're square looking, a little more durable, a little heavier of a lining. Columnar, in the digestive tracts where we see those, and we also see them in some mucous membranes. These oftentimes have the finger-like projections on the top of them too, because of the things that they're doing, but we'll see them columnars where we see them in the digestive tract. And then there's another classification called pseudostratified, which we see in the respiratory tract. They have a single layer, but some cells are shorter than others, giving a false impression of it being stratified when it's really not. You can kind of see where it kind of gives the impression that it's stratified, but it's really not, because they're just the way that they're there. And then we have stratified squamous, the mouth, esophagus, on our skin. These actually are good for protective layers, and is what their purpose is. So we can see that. Then we have stratified cuboidal, stratified columnar, and stratified cuboidal and columnar. So we find different types of these. I don't want you to get caught up in trying to go, oh, well, these belong there, and we'll find these there, and we'll find these here. Just realize, remember when I talked to you about all cells not being exactly the same shape and all that kind of stuff, that there's different shapes of cells? When we're talking about epithelial cells, we can see that there's a wide variety of different shapes and types and layers of these epithelial cells. And when we say epithelial, we're talking about like skin cells, membranes, those types of things. And there's different types of those. So don't kill yourself understanding them completely, but have a general understanding of them. You would need to know what a squamous cell looks like, and what a cuboidal cell looks like, and what a columnar cell looks like. All of those should be relatively easy. There is one other interesting one that is a transitional that we find mainly in the urinary tract and urinary system. It's in particular the bladder. It kind of looks one way, and then when we stretch it out, because it's full, it looks a different way. So it's kind of transitional looking, and that's why we call it that. It's because its shape changes according to how full it is. We can see that kind of looks that way there, and then it changes its shape when we stretch it. Also, we have glands that secrete things. That is also part of the epithelium, but it's also part of the exocrine system too, that we'll talk about later, the endocrine and exocrine system. But glands actually are present in that epithelial layer too, and they have different processes. Maybe they lubricate a surface, they might try to cool a surface, they might try to secrete something on top of that surface that protects us from bacteria and those types of things. There's two major gland types, and these are such major types, though they're under the epithelial area, we will talk about these in a separate chapter. One of them is the endocrine gland. Basically, it's endo. It means it excretes into. So that means that these particular glands will excrete directly into the blood vessels, directly into something. Hormones, thyroid, adrenal glands, pituitary glands, all those secrete directly into the bloodstream. Exocrine glands dump outside through ducts. So they may dump things into the digestive tract. They may be sweat glands on the surface of our skin. Those types of things. Those are exocrine glands. Those glands they're all over the place, as you can imagine, but they're highly specialized, and we'll talk a little bit more about them. Not the last time we're going to hear about them. Then there's also this connective tissue that's all over in the body. It's the most abundant. It provides protection. It connects things together. It supports the body. The one thing that seems the weirdest about the connective tissues, blood is classified as a connective tissue because actually it does connect to all parts of the body. Seems weird. So there can be some variations in blood supply depending on the tissue type and how the vascular needs, the oxygen needs of a particular tissue. The more oxygen demand that tissue has is the more vascular it'll be. Some have a poor blood supply, some have no blood supply. There are, let me back up here. There are certain things in the extracellular matrix, the things around cells, that are considered connective tissue. The two main elements of those are ground substance. It's mostly a watery, adhesive, kind of a sticky polysaccharide kind of substance, so it sticks to things. And then there's also fibers that are produced by the cells, and those fibers are collagen, elastin, and reticular fibers, which is a type of collagen. So those are the two main types of of extracellular connective tissue elements. And then we have some others, such as bone, cartilage, dense connective tissue, loose connective tissue, blood. Like I said, there's a lot of different types of connective tissue, and we're going to talk about each one of these in almost a separate chapter on their own. Bone and cartilage will be together. Blood will be together. We'll talk about dense and loose connective tissue when we talk about the body system over the skin. Of course, we'll talk about bone in particular. We'll talk about all these osteocytes and all those types of things. Don't get caught up in trying to remember all of these. We'll talk about cartilage later. I know it seems like I'm just kind of whipping through these, but I don't want you to focus and knock yourself out on studying things that we're not going to that I'm not going to test you on knowing them well just yet. We're going to talk about and test on them later. This is just kind of a welcome to, hey, guess what? This is a type of connective tissue, which is cartilage. And there's different types of cartilage. There's hyaline cartilage, which has a lot of collagen fibers that helps it with that rubbery feel to it. We find it in the larynx and the epithelial plates and those kinds of places. There's also elastic cartilage. There's fibrocartilage, several different types of cartilage. The elastic cartilage provides elasticity, just like it says. Fibrocartilage is highly compressible, and it gives some cushion. We find it between our vertebrae, even that little bit between our knees is another one of those places that we find it. Dense connective tissue, they maintain elements. The main element in them is collagen fiber. Tendons, ligaments in the dermis, we find it there. And then loose connective tissue, most widely distributed connective tissue, soft, pliable, usually like cobwebs. They soak up fluids and do all those kinds of things, and it's found everywhere, is that loose stuff. Another example of that is adipose tissue or fat tissue is a good example of that. It helps to insulate the body, hot and cold, protects from bumping and all that kind of stuff. Usually we have a layer of fat cells around different organs in the body to help protect them from shocks and bumps and all that kind of stuff. And it's a great site for fuel storage for future use. There's also, there's also some reticular connective tissue that's loose connective tissue. We find that in the lymph nodes and the spleen, and some of it even in bone marrow, that kind of holds those tissues together in those areas. Blood, oddly enough, is a connective tissue. It provides nutrients, disposes of waste, and one of the most important things that it does is its movement of those respiratory gases, which we'll talk about blood in depth at a later date as well. Okay? Muscle tissue, its function is to shorten and contract. Like I said, this is going to take us through each one of those elements or each one of those parts of the body. So I'm just going to kind of go through it pretty quickly because basically it's a couple of slides. I think it's maybe a paragraph or two in your book. I don't want to dwell on it. I don't want you to think that this is what's going to be important. The other stuff was what was more important. 3 different types: skeletal, cardiac, and smooth muscles. Skeletal muscles attach to bones and give us the ability to move. They're striated, meaning they have stripes. I talked about that a little bit earlier. We can see the striations in the picture there on your left. Cardiac muscles are involuntarily controlled. That's a good thing. We don't have to sit around and remind our heart to beat. Oh, hey, I forgot to tell my heart to beat. There would not be many of us, or probably any of us, still alive. Those that were still alive would only be sitting around thinking about their heart beating. So the cardiac muscle is striated too, and we'll talk about why it's striated later. Uninucleated, short branching cells, so they're a very unique cell. And we can see a picture of it there on the left and a real picture of it on the right. Smooth muscles are also involuntary. We find these in the digestive tract that moves food along our tract, also in our blood vessels. The walls of our blood vessels can be responsible for causing us high blood pressure and those types of things, but we can't control that. It's smooth muscle because there's no striations on it. Nervous system, there's neurons, all different types of neurons, and the way that those work are very specialized. To try to get into any of that right now would be really, really inappropriate. But we'll talk about the nervous system and the neurons and the central nervous system and the peripheral nervous system and all those different parts of the body. We'll talk about all of those in depth at a later date in later chapters. Our body has a great ability to be able to repair. They can regenerate. As we looked at this in the video of mitosis, he talked about it's essential that our cells are able to regenerate and rebuild and repair. Otherwise, we wouldn't exist. We wouldn't be here, or we wouldn't last that long if it couldn't regenerate and repair itself. Regeneration is the replacement of tissue. Fibrosis is the repair of dense fibrous connective tissue. Regardless of which one is being done, it depends on the type of tissue that's damaged and the severity with how long it will take for that repair to work or how well it will work. Such as a clean cut's going to heal better than a jagged cut. We probably all have a scar or two on us from a cut or something goofy that we did or some situation that we got ourselves in. And if it gets badly infected and those types of things, is going to increase the likelihood of that not being a clean heal or it taking much longer to heal than what it normally would. There are several events that happen during a normal tissue repair. We'll talk about these. These will be talked about a little bit more again, but the first part of that process is the inflammatory process. This is an intentional process that kind of protects that area and protects the rest of the body from that area as well, causes some swelling, blood clotting, walls that injured area off so it can't affect the rest of the body. Hopefully that's the ideal thing to do. That's what we're hoping for it to do. And then there's some granulation of the tissue that's going to start to happen. We'll get some new capillary growth, believe it or not. There'll be some new blood capillaries that'll grow into that area. And the reason for that is so that it can start to clean it up and start the repair process. We'll bring in some more phagocytes, which is part of the immune system to destroy the things that don't belong. Blood clotting will be there. Some fibroblasts will be there to start stitching things together. We'll have some collagen fibers that'll show up that'll work in there. And then we'll have, after that scab detaches, or the regeneration of the surface epithelium is indicated when the scab detaches. And whether or not there's a scar depends on how bad the wound was. And that whole process, like I said, it depends on how bad it was, is how long it's going to take. Most tissues will regenerate pretty easily. Or tissues that do, most of our epithelial tissues and our fibrous connective tissues and bone actually regenerate pretty easily. So it's not too bad there. Skeletal muscle, though, does not. It takes a long time to regenerate skeletal muscle. You wouldn't think so, but it does. And then there's some that are just completely replaced or nearly all replaced with scar tissue, just because the dividing ability of those cells is not very good. In particular, that's cardiac tissues that don't regenerate well, and nervous tissues almost don't at all. There is some evidence that they do to a small degree, but generally they don't. So as we grow throughout our lifetimes, cell division continues through puberty with many of the cells, and then some of that starts to trail off after puberty. Those areas of cell populations that are exposed to friction will replace, such as the epithelium replace or lost throughout our lifetime. It's interesting how many cells, skin cells, that we lose or replace in a year. The estimates are somewhere around 1 pound of skin cells that you shed off of your body throughout the course of a year, and maybe even more than that, I don't know. It sounds like a lot, but that's what you're leaving behind, shedding off all your skin cells. So if you let somebody stay the night at your house, that means you really like them, because they're going to leave their skin cells at your house. Connective tissue remains mitotic and forms repair tissue or scar tissue. So some of that connective tissue doesn't heal well. Muscles become amitotic by the end of puberty. And nervous tissue becomes amitotic shortly after birth. So we kind of have what we have with them, and it doesn't change a whole lot. Injuries can severely handicap those tissues, and the cause of aging is unknown, but chemical and physical insults, as well as the things that go on within our life, are in part because of that. It's a lot has to do with our environment. Some of it has to do with gravity. Some of it has to do with the things that we take in. It just comes with the territory. Not much we can do about it. There are is the possibility of what's called as neoplasms. They can either be benign or cancerous. Basically, it's just abnormal masses that grow within our bodies. There can be a cause like an you spent too much time in the tanning bed, or it can be just, we don't know why. It just happens. So it can be either or it can be that you smoked, 4 packs of cigarettes a day and now you've got lung cancer. We kind of go, yeah, well, that makes sense. But it's those times where you have a newborn that, or a 3-month-old that is diagnosed with a brain tumor. For us, that just, it's unfathomable, but it does happen. Hyperplasia is an example of what happens to tissue when we expose it or strongly stimulate it or irritate it. It begins to grow in size, increase in size. Atrophy is just the opposite of that. So hyperplasia, if you go to the gym and work out and you work your muscles, eventually the muscles will get bigger. That is an example of hyperplasia. Atrophy, on the other hand, if you break your leg and it's in a cast for 6 weeks and you take it out and it looks like somebody else's leg because it's a fourth of the size that it normally is, that's an example of atrophy because we didn't use those muscles in our leg. They decreased in size because they weren't being used. The body recycled those muscles and said, hey, look, you're not going to use them. We're going to use them somewhere else. And that's what happens and what we wind up with is atrophy. That, ladies and gentlemen, is chapter 3. I have almost lost my voice. It looks like we're right at 2 hours and 40 minutes. I do most often lecture via marathon. I'm a marathon lecturer, so that's not unusual for me to lecture that long. So that's it. I will have more for you guys on chapter 4.